English YouTube channel. How's everyone doing? Welcome to day 17 of your 50-day Neet challenge. How are you doing? Let me know in the comments if the audio and video are both clear. Hi Hishu, good morning. Hi Rohit. Hi Sham. Welcome to today's lecture. In today's class, we are going to be discussing all of the concepts in the chapter Respiration in Plants and also solve questions as we always do. I'm doing good, Rohit. Thank you for asking. Good morning, Kartikeya, Barik, Nikita, Shri Sham, Sneha, Akash. Akash L, he pretty much attends every lecture. Nikita, it's been 4 days actually. Yeah. Hi Vignesh, good morning. Hi Joy, good morning. How's everyone doing? Wait, I just read a quote that I think I should share with you. I was just scrolling through my WhatsApp and then someone I know had shared something very profound as his WhatsApp status update. I think you all need to listen to it also. Okay, so here it is. If you don't separate yourself from your distractions, distractions will separate you from your goals. So this is a gentle reminder for all of you to turn off all your mobile phone notifications that are distracting, all your WhatsApp notifications, Instagram, whatever social media app you're on. Turn it off because this chapter, you need to understand the concepts really well if you have to answer the questions based on this chapter. Okay? Hi Bireen, good morning. Channa Basava, good morning. Karnataka Dora Nivo. Amit Kumar, I'm doing great. Thank you. Hi Nikita, sending you all the love back from Bangalore. Okay, so should we get started with today's lecture? How many of you have already revised this chapter and have a fair idea of what we are going to learn here? Hi Maharaj. Okay, so respiration in plants, though this chapter we are learning under physiology, we've learned in the chapter Living World that cellular respiration is a characteristic feature of all living organisms, right? So bacteria, fungi, every living organism shows cellular respiration inside of its cells. So that's exactly what we are going to be studying in today's chapter. Let me quickly show you the syllabus. It's exactly like it was before, but there are a couple of things that are changed in this year's rationalized syllabus. When we get to it, I will tell you what exactly those changes are. All right. So we are going to learn about respiration, cellular respiration. What exactly does that mean? What are the different steps involved in it? There's glycolysis, anaerobic respiration, aerobic respiration. Under aerobic respiration, we learn about oxidative phosphorylation, Krebs cycle, electron transport system, all of that, right? And then a couple of smaller concepts are included in this chapter. Okay. Hishu, let's see. You've been waiting for this chapter, I suppose. Finally, you have your favorite chapter here. Let's see how well you will score. Okay, stay till the end and we'll see how well you'll answer the questions. Okay, is everyone ready? Give me a thumbs up and let me know in the comments if everyone's ready and pumped up for this session. Right now, I'm seeing 36 people. Hopefully, at the end of this session, there will still be 36 people or more. Yes, there is no deletion of concept, Srisham, but a couple of things are changed. Like I told you, when we get to that concept, I'll tell you what exactly it is. All right, Bunny Maharaj is full on ready. Let's get started with the trend analysis over the last few years, ever since 2016. If you take a look at the Neet exam papers, over the years, consistently 1 or 2 questions have been asked from this chapter. So you can expect like 8 marks from this chapter. It's a very interesting chapter. Many students find this very difficult, but hopefully after today's lecture, it'll be easy peasy, lemon squeezy for you all. So let's get started with the session. Whenever you may have seen how in certain interviews, celebrities are asked, what are the things that you can't live without at all? And then they say, I can't live without my lip balm, I can't live without phone, I can't live without internet and all of that. But if you think of it biologically, there are 2 things that we absolutely need at any cost in order to survive. What are the 2 things that we cannot live without at all? The 2 things that we cannot live without at all are food and oxygen. Of course, without food, fasting, you can live for a couple of days, but without oxygen, after a couple of minutes, we are dead, right? So why are food and oxygen so important? That's important for us to understand at the beginning. So one thing is, every one of our cells, living cells, metabolic reactions are happening, right? So metabolic reactions, some of them require energy, some of them produce energy. Right now, if you consider this class, I'm speaking. While I'm speaking, I'm moving my jaw, I'm moving my tongue. That is muscular movement. As you're listening, your brain is probably, your auditory nerve is sending messages to your brain, and your brain is trying to comprehend what I'm trying to say. Or you're probably blinking. That is again muscular movement. You're probably taking down notes. That's muscular movement. You've learned in the chapter locomotion and movement that muscles movement require energy. For your brain to function properly, it requires energy, right? Where do we get all of this energy from? Your answer would be food. It is a simple middle school kind of question. Where do we get energy from? We get energy from food. So it's clear that if you consume food, you get energy, and that will help you sustain, and that will help all the metabolic reactions happen, right? But why do we need oxygen? In the chapter breathing and exchange of gases, in the chapter body fluids and circulation, you learn a lot about oxygen, right? So we take in oxygen, it goes to our lungs, blood supplies it everywhere, and then brings it back, brings carbon dioxide back, and we exhale. So all of that, what exactly is the role of oxygen? Why do our cells need oxygen? That is the question we're trying to solve by understanding this chapter. The oxygen that we inhale, the oxygen that is transported to every one of our cells, basically breaks down food. Whatever food we've consumed, that food gets broken down and energy is released. That energy is what our cells will make use of. So if you think about it, there are several life processes that require energy. And the energy for those life processes can be obtained by the process of cellular respiration. So in cellular respiration, food is involved, oxygen is involved. So in one type of respiration, oxygen is involved. So food basically gets broken down, it gets catabolized, and in that process, some amount of energy is released. Right? Whenever something is getting built up, energy is consumed. Whenever something is broken down, energy is released. So in this process, we learn about how exactly the food that is present in, the energy that is present in food gets released in our cells. Is that clear? So far, is it clear? Hi Nandini, good morning. Hishu says yes, Anjali says yes, Akash, Rohit, so nice to see. Okay, awesome. Wait, let me recollect your name. You are Nagamuttu, right? I'll just call you Naga because it's easier. See, even after 5 days, I still remember your name. Okay, so let's get started. I've put up most of whatever important statements are there in NCERT on our slides. So in case you're using this as your notes for revision, then you're going to get all the important points from this PDF itself. Okay. So, like I mentioned earlier, life processes require energy. And how do we get energy? We get energy from the oxidation of macromolecules, which we call food. What are the macromolecules? In our food, what do we consume? We consume carbohydrates, proteins, fats, and all of that. So in our digestive system, it is getting broken down into the smallest monomer units, and then the blood carries it from our intestine to every part of the body. So through blood, our cells require obtain food, and through the same blood, our cells will obtain oxygen also. Within our cells, oxygen will break down this food and produce energy, and that energy is used by the cell for its metabolic activities. Okay, so talking about food, we have different groups of organisms that have different modes of nutrition, right? When you consider plants and certain algae and blue-green algae or cyanobacteria, they are photoautotrophic. They are capable of synthesizing their own food. But even though these plants are capable of synthesizing their own food, consider a mango tree or a banyan tree. Do every cell in the banyan tree have the ability to photosynthesize? Absolutely not. In order to perform photosynthesis, chlorophyll is required. But where is chlorophyll present? Only in certain cells of plants, right? So it's present mostly in the leaves. And it's present probably in young stems. But there are several cells even in plants which do not have the ability to photosynthesize because they do not lack the enzymes and the pigments that are required for photosynthesis. So even though some cells of plants photosynthesize and prepare food, there are cells that cannot do so. Even within plants, cells that prepare food transport it to cells that do not prepare food. Okay? Yeah. Akshay, I think your question is answered. Rohit has answered it like the class leader. Okay? So in photosynthesis, how is food synthesized? Sunlight, there's some energy present in sunlight. That energy is trapped by your pigments. Carbon dioxide is taken into the plant cells. Carbon dioxide is combined to form glucose and other such molecules, right? So that is photosynthesis. Plants, clear. What about animals? How do you and I obtain food? Obviously, we do not have the ability to synthesize it within our own body. We obtain it directly or indirectly from plants. If you're a vegetarian like me, you will directly obtain it from plants. If you're a non-vegetarian, unlike me, then you will obtain it indirectly from animals that eat plants. Okay, so this is your heterotrophic mode of nutrition. And then we have certain groups of organisms called saprophytes. These saprophytes depend on dead and decaying matter. Okay, so they obtain their food and they derive their nourishment from, let's say, dead plant matter or dead animal matter and all of that. So they are called saprophytes. Every organism has a different way of obtaining nutrition. Yeah, right. Ultimately, everything is dependent on plant matter either directly or indirectly. Whenever photosynthesis happens, glucose is produced, right? Glucose is the end product of photosynthesis. One of the products of photosynthesis is glucose. In glucose, you have 6 carbons. Okay, Savita, this is confusing. Yes, Rohit, that is in ecology, ecosystem. We learn about pyramid of energy. There, that is the concept we will discuss. For now, let's focus on respiration. Okay, in whatever I'm saying right now, because we have little time, we have to solve a lot of interesting questions. I'm saying it very fast. Hopefully, this will be your revision and you're able to keep up. If in case you have any doubts, even in the middle of this, you can kindly let me know. Okay. So, yeah, whatever substances our cells make use as food, whatever molecules our cell use as substrates for cellular respiration, they are referred to as respiratory substrates. So basically what happens during respiration is, in all of the food molecules that we consume, be it carbohydrates, be it proteins, be it fats, there is carbon-carbon bonds, right? There are many carbons that are linked by bonds. The energy is present in these bonds that are present in this carbon skeleton. What happens during the course of respiration is this complex compound will get oxidized. When the complex molecules, be it carbohydrates, proteins or lipids, when they get oxidized, some amount of energy is released. Now these complex compounds, like I already mentioned, could be carbohydrates, proteins or fats. But in most cases, our cells will prefer carbohydrates to be the preferred substrate. But in certain cases, under certain conditions, even proteins, fats and organic acids also can be used as substrates. Okay, so most common, most preferred is carbohydrate. Rarely, under certain conditions, even proteins, fats and organic acids can be used as respiratory substrates. Alright? Now, how exactly is this energy released? How exactly do our cells make use of it? That is something that we need to understand. Hi Venkat Pushpa, good morning. Nan super agidini, neevu hegidira? Yeah, I was actually thinking why Venkat Pushpa has not dropped a message today. Hi Sagar, you're late for the class. So, yeah. What exactly happens here is, let me give you a very relatable example. Let's say I'm working for Physics Wallah, I'm taking classes every day and for that I get paid. Okay Sagar, be on time from the next class. For that I get paid at the beginning of every month. That is, you can think of the energy that is present in a molecule. What I do is, I do not use all of it on the same day. I utilize it, I save it such that whenever I need it, I can make use of it. Okay. So if I'm getting some amount of money, I do not spend it all on the same day. I save it so that I can spend it whenever I need it. So if I have to, for example, take a cab to the studio for recording, then I will spend it. If I want to eat a gulab jamun, then I will spend it. But I do not spend it all on the same day. Yeah, I'll get to that. I'll get to that, Chandu, in just a bit. I'll tell you the differences between breathing and respiration. So, Shant, good morning. Welcome to the lecture. Yeah. So, even in our cells, if you consider our cells, there are anabolic reactions happening. There are reactions happening where something is getting built up, and there are catabolic reactions happening where something is getting broken down. What we basically do here is, for anabolic reactions, whatever energy is needed, we try to get it from the catabolic reactions. So, if something is getting broken down, obviously a lot of energy is going to be released. Instead of letting that energy be lost as heat, we make sure that that energy that is being released is trapped in molecules called ATP. So, this ATP is like a reserve of energy. Whenever the cell needs energy at any point in time, it can make use of ATP. So ATP basically looks like this. Okay, everybody has studied molecular basis of inheritance, right? You all know what is ATGC. Adenine, guanine, cytosine, thymine, all of these are nitrogenous bases. You know about that. So ATP is nothing but a nucleotide. It is a nucleoside triphosphate. Here you have the sugar. Here you have the nitrogenous base adenine. To it, generally, when you consider a nucleotide, to the 5' and 1 phosphate will be attached, but here 3 phosphates are attached. Because 3 phosphates are attached, we call it adenosine triphosphate. Over here in adenosine triphosphate, this bond right here, in between the second and the third phosphate, is a high energy bond. What that basically means is, whenever this bond is broken, a lot of energy is going to be released. Okay, so what the cell does, right now, something is getting broken down. That breakdown reaction is going to release energy. If the cell does nothing about it, that energy will be lost as heat. But what can we do instead? Later, something might get synthesized in the cell. At that time, we need energy. Right now, something is getting broken down, that is releasing energy. So, the cell will very cleverly save whatever energy is released during breakdown in molecules called ATP. So, this ATP can later be used while something is getting built up inside the cell. How to overcome exam fear? Chandu, first of all, you need to identify what is the reason for your fear. Is it because you are not prepared enough? Is it because you're feeling like you're not prepared enough despite preparing well? Or you faced failure in the past and you're afraid that it will repeat? What exactly is the reason you are experiencing fear? That you have to identify. Once we get to the cause of why you're feeling fear, we can find out answers and solutions for it. Because Karthik will help you understand the concepts very well. Yes, Prithvi, phosphoester bond. Okay, so what happens is, whenever the energy is getting liberated, a molecule of ADP, this is A containing 2 phosphates, that's why we call it adenosine diphosphate. Di means 2, right? Adenosine with 2 phosphates is adenosine diphosphate. This adenosine diphosphate combines with another phosphate, inorganic phosphate, to form adenosine triphosphate. So, energy is stored in the form of ATP. Is that clear? Is whatever I am trying to say so far clear? In our cells, breaking down is happening. In our cells, building up also happens. While breaking down, energy is released. If you do nothing about it, energy will get wasted. What we do instead is, when the energy is released, of course, all of it cannot be trapped into ATP. How much ever is possible, we try to trap that energy in the chemical bonds of ATP. We use that energy to synthesize a molecule of ATP by combining ADP and inorganic phosphate. Whenever in the same cell there is a need for energy, this ATP will get broken down. When ATP breaks down, undergoes hydrolysis to form ADP and inorganic phosphate, some amount of energy is released. That energy will be used by the anabolic reactions. Akshay is asking how to obtain notes. After this class is over, whatever PDF I'm displaying right now, whatever PPT I'm displaying right now, this will be available to you as a PDF in the description box. Okay. So it will take about 1 or 1.5 hours after the class to upload it. So you can find it there, you can download it. All right. Now that this is clear, let's talk about an important question. What is breathing? What is respiration? Who can tell me the difference in the comment section? Are breathing and respiration the same thing? You've already learnt about breathing and exchange of gases in human physiology. Who can tell me the difference between breathing and respiration? Breathing is a physical process where you take in oxygen, give out carbon dioxide. Respiration includes breathing. So if in me respiration has to happen in my cell, cellular respiration has to take place, then breathing is a part of it. I breathe in oxygen. The oxygen from my lungs is transported to my blood. My blood will carry it to my cells. Their cellular respiration will happen. As a product of cellular respiration, carbon dioxide is released. That enters my bloodstream. Carbon dioxide is transported back to my lungs and I exhale it, right? So inhaling, exhaling, that is breathing. Respiration includes breathing, but respiration and breathing are not the same thing. Yes, Venkata Pushpa is right. Respiration is a process where food breakdown takes place. Basically, oxidation of food takes place. Oh my God, Sri Sham has given like a proper definition. Please tell me you didn't copy this from Google. All right, most of you have somewhat gotten it correctly, so I'm proud. I'm happy that you've understood it well. All right, so do plants breathe? Do plants require oxygen in the first place? If plants have to perform cellular respiration, just give me a moment. Okay. Yeah. So do plants require oxygen? We generally say plants take in carbon dioxide, give out oxygen. That is true with respect to photosynthesis. During photosynthesis, carbon dioxide is taken up by plants and they perform photosynthesis, produce glucose and oxygen, and the oxygen is released. For photosynthesis, carbon dioxide is taken in, oxygen is released. But do plants need oxygen at all? Similar to every organism, plants also have to perform cellular respiration, otherwise there's no way it can obtain ATP, right? You know how for photosynthesis ATP is so required, right? So there are many steps in Calvin cycle where ATP gets consumed. Where does that ATP come from? That ATP comes from cellular respiration itself. So even plants require oxygen, but they do not breathe. And we have elaborate respiratory system, we have respiratory organs, lungs and all of that. But plants don't have any of those. Despite not having elaborate respiratory organs, plants still somehow manage to survive. How do they do it? So that is the question we need to answer here. There are 3 reasons because of which plants can survive even in the absence of specialized respiratory organs. The first important point here is each part takes care of its own gas exchange needs. You know that there are openings present in pretty much every part of the plant, right? In the thick tree trunks, you will have lenticels. In the leaves, you will have stomata. And in younger stems, you will have stomata. So there are openings to the exterior that are present in every part of the plant. That is not the case for us. If my feet needs oxygen, it has to go to my lungs and to the blood and then go to my feet. But that's not the case in plants. Every part pretty much takes care of its own gas exchange needs. That is why there is no elaborate system required for it. Second important point, plants do not present great demands for gaseous exchange. We require a lot of oxygen because the rate of respiration in animals is much higher compared to the rate of respiration in plants. Also, majority of gaseous exchange happens in plants during photosynthesis. For respiration, not much oxygen travels from the outside, from the environment into the plant because photosynthesis is anyway happening. As an end product of photosynthesis, oxygen is anyway getting produced, and we need oxygen for cellular respiration. That's right there in the cell. It can make use of that oxygen itself for cellular respiration. All right. So, Srishti, hi, welcome to the lecture. I think you joined a little late. The weightage, you can expect like 1 to 2 questions. I would say 2 questions from this chapter. I always discuss the weightage in the beginning of the session. Okay, so what basically happens here is, so oxygen is not even an issue here because plants are photosynthesizing. Oxygen will anyway be produced as a result of photosynthesis, as a product of photosynthesis that the cells can use for cellular respiration. The third most important point is the distance that gases must diffuse is not great. Why is that so? If there is a tree trunk that is so huge, how can we say that gas need not travel a long distance? Of course, in leaves I can understand because leaves are very thin, stomata is present, and diffusion not long distance. But if it's a tree trunk, then what is the case? If you think about it, you know that the center of the tree, you've studied about secondary growth probably last year, and you know that in the center of the tree you have heartwood where there is no living material present at all. Only in the surrounding peripheral area you have living cells and tissues present. So even if you remember anatomy of flowering plants, which I myself taught you in this 50-day challenge, you know that the cells in the cortex are loosely arranged. So there's a lot of intercellular spaces between the cortical cells, between the parenchymatal cells in the cortex. So there's not much distance that gases will have to travel in order for respiration to take place. Okay, so these are the reasons why, despite not having elaborate specialized respiratory organs, plants can still survive. Okay. Hishu, behave yourself. What 2 lines did you not understand, Soumya? If you could be a little bit more specific. Like these 2 points you didn't understand. Yeah, Sagar is right, heartwood. But this year that has been deleted from your syllabus. Is that clear? Soumya, what did you not understand? Since you're not answering, I assume you have understood. I'll move forward. Okay. So, like I already mentioned, if glucose is undergoing combustion, if it's getting broken down, then the complete combustion of one molecule of glucose, which produces, okay, this should have been a superscript, I'm sorry, subscript, but I don't know why, there is a typing error. This is carbon dioxide and water. It will, when it's broken down completely, it will yield energy. Like I mentioned earlier, most of it will be lost as heat. But what does the plant do? This we're learning under plant physiology, so I'm emphasizing plant. But that's what will happen in every living organism. This is not specific to plants, okay? Just give me 1 minute, Sneha. Since I've started this, I'll finish this off and I'll quickly go back and explain those 3 points. Okay? So what they do is, they have a strategy where they will break down this glucose in very small steps, such that each step is just small enough so that one ATP molecule can get synthesized out of it. How exactly it happens, we'll learn later. Okay, so carbon dioxide in aerobic respiration, I'm emphasizing oxygen is required only in aerobic respiration. Even in, there is a type of respiration called anaerobic respiration, over there oxygen is not required. Okay, if glucose has to completely get oxidized, then oxygen is required. So glucose, okay, battery is low. Glucose gets oxidized, gets broken down by making use of oxygen. In this process, carbon dioxide gets released, water also forms, and some amount of energy is released. What we are basically trying to do here is, we are trying to trap some amount of that energy in the form of ATP. How much ever energy glucose has entirely cannot be trapped as ATP. Some amount definitely will be lost as heat. A portion of the energy that is liberated is what we try to trap as ATP so that we can use it later. Okay? Uh, Sneha, we'll get to the points. Poll, I'll start in just a bit. Soumya, are you still there? I'm going to explain the points again. Okay, so the points that we're trying to understand here is we have respiratory organs, plants don't. How is it that plants can still survive well even in the absence of respiratory organs? First of all, every part of the plant takes care of its own needs. Like I mentioned before, if my feet has to receive oxygen, the feet itself cannot receive oxygen. It has to go through my nostrils, to my lungs, then to my blood and then to the legs. That is not the case in plants because every part of a plant will have apertures either in the form of stomata or in the form of lenticels. So trunk will take care of its gaseous exchange, leaves will take care of its gaseous exchange. Every part pretty much takes care of its own gaseous exchange needs. The second point here is that the rate of respiration comparing plants and animals is very, very different. Animals respire at a much higher rate compared to plants. In plants, the rate of respiration is less, and in plants, photosynthesis also happens. Cellular respiration also happens. For photosynthesis, plants take in carbon dioxide from the atmosphere. This everybody knows, in the previous chapter also we've discussed, right? So plants take in carbon dioxide, and in the process of photosynthesis itself, oxygen is produced. So plants need not take a lot of oxygen from the atmosphere for cellular respiration. It's directly available to them right there. So majority gaseous exchange happens in plants for photosynthesis and not for cellular respiration. And the third more important point is that even in very large plants, the distance that gases have to travel, the distance that gases have to get diffused is very less. Because if you consider leaves are very thin, there are very few layers of cells, diffusion distance will be less. But even if you consider a thick trunk of a mango tree or a banyan tree, if you see, the center part of the trunk is dead tissue. You have heartwood there, which is not very useful. It does not have any living cells. If they are not living cells, there is no need of oxygen. The living cells are present mostly in the outer layers, in the peripheral layers. Over there also you have cortex, and this cortex has cells, parenchymatous cells, that are loosely arranged, and in between that there is a lot of intercellular spaces. So the gas from outside, the oxygen or carbon dioxide from outside, does not have to travel like a really long distance. It will be a short distance itself. I hope it is clear. Who had asked? स्पैम करने से समझ आ जाता है क्या अभिषेक? Rudra, please stop spamming. Whenever we are discussing questions, we'll be doing the poll. Okay, so let's move on. So the point here is, we have different types of organisms. Some organisms need oxygen to survive. Like we are aerobic organisms, we don't get oxygen for a few minutes, then we will die because our body is not getting oxygen to prepare ATP. If ATP is not there, then we cannot function properly, right? That is, that everybody knows. So, there are organisms in the world which exist, which cannot survive in the presence of oxygen. If you remember in the chapter Evolution in 12th grade, you've learned about when the living organisms originated on Earth. When was the first living cell formed? It was formed at a time when in the atmosphere there was not enough oxygen. In fact, there was no molecular oxygen at all. Those organisms have survived even in the absence of oxygen. To this day, we still have descendants of those organisms which are anaerobic in nature. They are anaerobic, meaning they cannot survive in the presence of oxygen. Okay, so if there are organisms that cannot survive in the presence of oxygen, then how do they synthesize ATP? Because I've been saying since the beginning that for ATP generation, for ATP synthesis, oxygen is required. But these organisms, they die if there is oxygen. How does cellular respiration take place in them? How do those organisms produce ATP? That is a question we have to answer here. So over here, what happens is, in the process of cellular respiration, there is one step which is anaerobic. There is one step which does not require oxygen, and in that step also ATP will be produced. So in those organisms which are anaerobic, they will obtain energy by that first step which does not require oxygen. Whatever steps require oxygen, those steps do not take place in that organism. But we must understand one thing very clearly here. whenever oxygen is involved in respiration, the amount of ATP we get is very high. Whenever there is no oxygen involved, it's happening anaerobically. How much ATP is produced is much, much lesser. How much less exactly, we'll get to the numbers towards the end of the session. Try to understand here, in aerobic and anaerobic, there is one step which is common to both. And that step is anaerobic. That does not require oxygen. There might be organisms which die in the presence of oxygen. Even in them, respiration will occur anaerobically and produce a small amount of ATP. Is that clear? Obligate means Sagar. Okay, I'll explain what facultative and obligate is. Obligate means 100% anaerobe. That means if there is oxygen, it will die. That is obligate anaerobe. We are obligate aerobes. We require oxygen to survive. In the absence of oxygen, we will die. Obligate means absolute requirement. But facultative aerobe means I require anaerobic conditions, but in the presence of aerobic environment, I will not die, I can somehow manage. Okay, yeast is an example. It does not always require anaerobic conditions. We give it anaerobic conditions to obtain alcohol from it, to obtain, for baking industry and all of that. But it can survive in aerobic conditions as well. Obligate means very strict. We say viruses are obligate parasites. What does that mean? They will survive very strictly only when a host is available. Outside the body of the host, they are as good as dead. That means they very strictly need that. Okay. I hope it is clear. Let's move on. Yeah. So all living organisms, regardless of whether they are aerobic, anaerobic, facultative, obligate, they will have the enzymes that are required for that first important step of respiration. All living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This is important. If it is anaerobic, oxidation is only partial. Complete oxidation of glucose will not take place, and also energy that is trapped, ATP that is produced, will be in very small numbers. Is it clear so far? Is it clear? So that step, that first step, which is anaerobic and where partial oxidation of glucose takes place, that is called glycolysis. Actually, it seems like a very small one-step reaction, but glycolysis actually has 10 steps, and these 10 steps, it's very important for you to memorize. So I'm thinking of coming up with a song for glycolysis so that you all can remember it very well. Hopefully in the next 4 or 5 days, I will post it on YouTube as a strategy song. So yeah, glycolysis is universal. Every living organism, you can see glycolysis. Okay. So how many of you here know the song Despacito? It was popular a few years ago, probably about 10 years ago. I'm not really sure. How many of you know the song Despacito? It's a Spanish song. I'm trying to make make a song for glycolysis in the tune of Despacito so that you will never forget the steps of Despacito. Okay, Soumya knows the song. Shri Sham knows the song. Yeah, it's pretty popular. Okay, so you will get to hear it in a few days. Okay, so that should help you, remember all the steps of glycolysis. All right. Now, why are we calling this first step glycolysis? Glyco means what? Glyco means sugar. Lysis means what? Splitting or breaking down. In glycolysis, there's a partial oxidation. Yes, Srisha. There is partial oxidation of glucose where glucose is being split. Okay. So this glucose, everybody knows glucose has 6 carbons. This 6 carbon molecule splits into 2 molecules containing 3 carbon, and because of this partial oxidation, some amount of energy is released. Okay. So that is your glycolysis. Over here, I've represented it in just one equation, but it actually takes 10 steps. Glycolysis has an alternate name. You've learnt about several cycles even in photosynthesis in the previous botany lecture, right? Chaitra ma'am's lecture. She's taught you about cyclic photophosphorylation, non-cyclic photophosphorylation, and then you have Calvin cycle. Calvin cycle is also called C3 cycle. Then you have C4 cycle. So you have different cycles. Sometimes one pathway will have more than one name, and you should remember the alternate names as well, because they will not always ask you questions saying that this is glycolysis and these are the steps, which one is incorrect, right? Sometimes they might ask you indirectly, so you should know the alternate names of every pathway. So glycolysis is also called EMP pathway, similar to why other cycles are called Krebs cycle and Calvin cycle, named after the scientist. The people, there are three people who first elucidated glycolysis. To give them regard and respect, we also call glycolysis as EMP pathway. Who are these three people? Extremely strict looking people. So there's Gustav Emden, and then there's Otto Meyerhoff, and then there's J. Parnas. Okay, so these three guys, they elucidated the pathway of glycolysis. So paying respect to their contribution, we call it the EMP pathway. Okay, so EMP pathway is Emden, Meyerhoff, Parnas. What a way to remember, Sagar. Okay, so this is basically the first step of cellular respiration. Like I already mentioned, this happens in every living organism. Like somebody pointed out earlier, it is universal, right? It happens in all living organisms. In anaerobic organisms, this is the only step that will happen. Whatever happens later will require oxygen, and that will happen only in aerobic organisms. Where does this take place? Site of glycolysis is the cytoplasm. Over here, I'm repeating again, glucose is not completely oxidized, it's only partially oxidized, right? Okay. All right. So, since this chapter we are learning under plant physiology, where do plants get glucose from to respire? So plants are capable of photosynthesis. They synthesize glucose. If the glucose has to be transported, it is transported by means of sucrose, which is a disaccharide, or if it has to be stored, it's stored in the form of starch, which is a polysaccharide. So within the plant, glucose as it is could be present. It could be present as a disaccharide in the form of sucrose, or it could be present in the form of starch. Hi Kajal, good morning. Welcome to the lecture. Okay, so if sucrose is present, sucrose, like I mentioned, is a disaccharide. It will have two units. One unit is glucose, the other unit is fructose. In the first step of glycolysis, an enzyme called invertase catalyzes the breakdown of sucrose or the splitting of sucrose disaccharide into two monosaccharides, glucose and fructose. This glucose and fructose will readily enter the glycolytic pathway. Is it clear so far? Now let's begin with glycolysis. Please pay attention. This is very important. There are 10 steps. Exactly what happens where you need to understand. Glucose is the most preferred substrate. It is still not the smallest form, right? The smallest unit is a monosaccharide. So that is the most preferred one. Glycolysis itself means we're breaking down glucose, right? Is it clear so far? Is everybody ready? Yes, everybody enter into the world of glycolysis. Right now, as we are speaking, our cells will be performing glycolysis. Okay, everyone's ready? All right, let's get started. Where did we get this glucose from? We got it from sucrose. Invertase acts on sucrose, breaks it down into glucose and fructose. Glucose and fructose directly enter the glycolytic pathway. We're starting the glycolytic pathway with glucose. Everybody pay attention. I want 100% undivided attention, extreme focus. Okay? Because if you don't get one step somewhere here, you will not understand the steps here. Okay? So we're starting with glucose. In glucose, how many carbons are present? There are 6 carbons. What did I say happens in glycolysis? A 6-carbon molecule of glucose will produce 2 molecules of a 3-carbon compound called pyruvic acid. These are the only 2 things that I mentioned, right? But in order for this to happen, 10 steps will take place. Of these 10 steps, in the first 5 steps, ATP will get utilized. It's like it's preparing. It's called the preparatory phase. In the next 5 steps, steps 6 to 10, ATP will get synthesized. The whole idea of performing glycolysis is to trap the energy that is present in the bonds of glucose, right? So we are partially oxidizing glucose, splitting a 6-carbon molecule into 2 3-carbon molecules. This happens in 10 steps. Over the course of this process, some amount of ATP is, yes, glucose activation phase, some amount of ATP is produced. Okay, ATP and another molecule also is produced. I'll get to it. So in the first step, what happens? Glucose has 6 carbons. Glucose gets phosphorylated. That means phosphate gets added to glucose at the 6th carbon. Glucose has 1, 2, 3, 4, 5, 6 carbons. At the 6th carbon, phosphate will get added. And glucose will form glucose 6-phosphate. This is 6 carbon. This also has 6 carbon. But the difference between this and this is that in this there is no phosphate. Over here at the 6th carbon, you have a phosphate attached. So for the attachment of phosphate, where did the phosphate come from? In the first step, a molecule of ATP will get broken down into ADP and inorganic phosphate. This inorganic phosphate is added to the 6th carbon of glucose so that glucose will produce glucose 6-phosphate. Next step is isomerization. Isomerization, you know, glucose and fructose are isomers, right? So they have the same chemical formula, but the structure is slightly different. The glucose 6-phosphate that is formed in the first step will undergo isomerization, change its structure and become fructose 6-phosphate. Over here, there is one 6-carbon compound, 1 phosphate. Here also there is one 6-carbon compound, 1 phosphate. There are 6 carbons and 1 phosphate. There are 6 carbons and 1 phosphate. There is no change in the number of carbons. There is no change in the number of phosphate as well. Okay. Yeah. So, next step what happens? Fructose, which has 6 carbons, to the 6th carbon 1 phosphate group is attached. In the next step, to the 6 carbon fructose, to the first carbon also another phosphate will get attached. Okay. So fructose 6 phosphate will form fructose 1,6 bisphosphate, where at the first carbon there is a phosphate and at the 6th carbon also there is a phosphate. Fructose has 6 carbons, but right now it has 2 phosphates. 1 phosphate it already had at the 6th carbon. Where did the other phosphate attached to the first carbon come from? Again, another molecule of ATP gets utilized, it gets broken down and form ADP plus inorganic phosphate. This inorganic phosphate is attached to the first carbon of fructose 6-phosphate and it becomes fructose 1,6 bisphosphate. Okay. So, over here you have to remember the name of an enzyme. In the first step, the enzyme involved is called hexokinase. Okay. So, wherever enzymes are there, remember that. So, previously we learnt about invertase. Remember this enzyme also. And this is hexokinase. This is the only enzyme we are going to learn about in glycolysis. Some textbooks, some coaching material will give you enzymes for every step, but that's not required. All there is in NCERT is sufficient. Okay. If glycolysis has to happen in sucrose Anshul, again the same thing will happen. Sucrose is the starting point, right? Glucose will not be stored or transported as it is. It is the end product of photosynthesis, but it will not be used as it is. So for storage, it will be present in the form of starch or sucrose. If it is in the form of sucrose, invertase will act on it, it will break it down, then glucose will enter this pathway. That is a choice, but why do you need to bombard your head with extra information when it's not required, Sagar? As it is, you have so many things to learn. Why do you want to burden yourself with information that will not help you in the exam? It's good, knowledge is never going to go waste, but is it necessary really? That is a question. Sebastian. Annie's marks are not increasing. Okay, let's get to how you can increase your marks in a little bit. For now, let's focus on the next steps of glycolysis. How many steps are over so far? First step, second step, third step, 3 steps we finished. In the first step, we saw ATP was utilized. In the second step, nothing really major happened. In the third step, again ATP is utilized. What is the fourth step? The fourth step, this fourth step, what happens is, this fructose, which has 6 carbons, will split into 2 molecules, each containing 3 carbons. This has 3 carbons. This also has 3 carbons. It has 6 carbon and 2 phosphate. It's splitting right now to produce 2 molecules, each containing 3 carbons and 1 phosphate. One of them is glyceraldehyde 3-phosphate. It's called G3P or PGAL also. How is PGAL related to this? Glyceraldehyde 3-phosphate is also called phosphoglyceraldehyde, so PGAL. The other one is DHAP, dihydroxyacetone phosphate. Dihydroxyacetone phosphate is also a 3-carbon compound that contains 1 phosphate. Okay, so here we have 6 carbons, 2 phosphate, splitting to produce 2 molecules, each containing 3 carbons and 1 phosphate. Okay, which are the 2 molecules? PGAL and DHAP. PGAL is short for phosphoglyceraldehyde and DHAP is short for dihydroxyacetone phosphate. Similar to how glucose and fructose are isomers, DHAP and PGAL are also isomers. So DHAP and PGAL, they are isomers. DHAP here in the next step will get isomerized to PGAL. So from the next step onwards, we will have 2 molecules of PGAL. Is that clear? Is that clear? This is the first part. Is it clear so far? In your NCERT, it's given continuously, but it's the same thing that I've continued here because clearly there is no space on the board. So, right now what happens? This will get converted into this. So, I have written here 2x. 2x means there are 2 molecules of this. That's what it means. From here, from this point onwards, there are 2 molecules of each. Because this also got converted into this. In the beginning of the lecture, I mentioned that partial oxidation will happen. Right? Partial oxidation will happen. Has partial oxidation happened so far? Has in any of these steps have oxidation happened so far? It hasn't. In this step, oxidation will happen. This is a very, very important step. So keep this in mind. A lot of things are going to happen in this step. 3 very important things are going to happen. We have PGAL here. PGAL is nothing but your phosphoglyceraldehyde. Right? We have 2 molecules of phosphoglyceraldehyde. In this step, glyceraldehyde is going to get oxidized into acid. Phosphoglyceraldehyde, it has 3 carbons and 1 phosphate. It is going to get oxidized into a 3-carbon compound called phosphoglyceric acid. As glyceraldehyde gets oxidized to acid, phosphoglyceric acid, a molecule of NADH plus H plus will get synthesized. Over here what happens is PGAL will provide or donate 2 redox equivalents. I'll write it here. Oh, what have I done? How do I go back? Okay. 2 redox equivalents is provided by PGAL. One of these redox equivalents is a hydrogen ion proton. The other one is a hydride ion. What basically happens is NAD plus will combine with hydride ion and become NADH. This other H plus will remain as H plus. That is why you have NADH plus H plus. Phosphoglyceraldehyde molecule is providing 2 redox equivalents, one in the form of a hydrogen ion, the other one is form of a proton, the other one is in the form of a hydride ion. So this NAD plus will accept the H minus and become NADH and the other H plus will remain. That's why we write it as NADH plus H plus. I hope this is clear. So in this step is where oxidation is happening. Okay. So phosphoglyceraldehyde is getting oxidized to acid, while here oxidation is happening simultaneously. NAD plus will get reduced to NADH and H plus. Is that much clear? That is the first important thing that is happening. Okay, so oxidation is happening here. When we say glycolysis is oxidation, we are talking about this oxidation here. Second thing, in glyceraldehyde 3-phosphate, you have 1 phosphate. In 1,3 bisphosphoglyceric acid, you have 2 phosphates. One at the first carbon, the second at the third carbon. Where did that extra phosphate come from? Over here, ATP will not get split, ATP will not get utilized to form, to provide a phosphate. Instead, phosphoric acid will donate the phosphate group to 1,3 bisphosphoglyceric acid. Anuj, this is a pure English channel, so we will be conversing mostly in English. Anshul, yes, in both aerobic and anaerobic, glycolysis will take place. Anaerobic degradation, Srisham, is because over here right now oxidation is happening, but oxygen is not yet used, right? This is a process common to both aerobic and anaerobic. This process in itself does not use oxygen, so it is an anaerobic process. If it is anaerobic respiration or aerobic respiration, in both the first step is glycolysis. In anaerobic, it's over, right there, it is over, okay. In aerobic, whatever the next steps are there, over there oxygen will be utilized. But in this process, oxygen is not utilized. Is that clear, Srisham? Any more doubts? Day 2 video, it's not there in the playlist. I'll find out. I'll get it updated. I have to check. Okay, everyone's understood? Alright. So this is the most important step. 3 things. Aldehyde to acid, oxidation, NAD plus to NADH reduction. You have 1 phosphate here, you have 2 phosphates here. The extra phosphate came from phosphoric acid, which is your H3PO4, I think. Right? So here you have phosphoglyceric acid. In the next step, what happens? 1,3-bisphosphoglyceric acid. It has 3 carbons and 2 phosphates. This will form 3-phosphoglyceric acid. So in 3-phosphoglyceric acid, you have 3 carbons but 1 phosphate. Where did that extra phosphate go? That extra phosphate is utilized in the formation of ATP. So ADP will take that inorganic phosphate from the first carbon, combine it to the ADP and form ATP. Here we have 3-phosphoglyceric acid that next converts into 2-phosphoglyceric acid, or you can say 2-phosphoglycerate. In 2-phosphoglycerate, you have 3 carbons again, and at the second carbon, you have 1 phosphate. The difference between this and this is in the position of where phosphate is attached. In 3-phosphoglyceric acid, phosphate is attached to the third carbon. In 2-phosphoglyceric acid, phosphate is attached to the second carbon. Both of these are basically positional isomers. Here also isomerization reaction will take place. In the next step, 2-phosphoglyceric acid gets converted into phosphoenol pyruvate. Phosphoenol pyruvate also has 3 carbons and it has 1 phosphate. So over here there is no change in the number of carbons, neither there is change in the number of phosphate. But one important thing you should remember in this reaction is a molecule of water gets released. In the final last step of glycolysis, phosphoenol pyruvate has 1 phosphate and 3 carbon, but pyruvic acid will have 3 carbons and 0 phosphate. Where did that extra phosphate go? It is in the synthesis of ATP. The phosphate here will get added on to ADP and form ATP. Is that clear? याद कैसे होगा दिव्या? I have thought about making a song for this. I will post it very shortly. हर्ष क्या explain करना है एक बार और? Let's revise it all entirely. Okay. So first we are starting off with sucrose. Sucrose is a disaccharide catalyzed by enzyme invertase, gets broken down into glucose and fructose. Both of these will enter the glycolytic pathway. Let's start off with glucose. Glucose has 6 carbons. A phosphate gets added to the 6th carbon of glucose and it forms glucose 6-phosphate. For this, an enzyme hexokinase is required. Hexo means it's a 6-carbon compound. Kinase basically is about wherever ATP is forming, kinase enzyme. That is the terminology we use. Okay. So in the first step, one ATP is getting utilized. Next step, what happens? Glucose 6-phosphate, fructose 6-phosphate, they are both isomers. Glucose 6-phosphate gets converted into fructose 6-phosphate. The number of carbons, the number of phosphate, both are the same. There's no change. In the next step, fructose 6-phosphate gets phosphorylated and forms fructose 1,6 bisphosphate. At this moment, to the first carbon also another phosphate gets attached. Where did that phosphate come from? It came from the splitting of ATP. ATP gets hydrolyzed and become ADP. That extra phosphate is attached to the first carbon of fructose. In the next step, fructose 1,6 bisphosphate, which has 6 carbons and 2 phosphates, will split to produce 2 molecules, each of them containing 3 carbons and 1 phosphate each. Okay, so these are PGAL and DHAP. Similar to how glucose and fructose are isomers, DHAP and PGAL are also isomers. At this point, the DHAP will isomerize and become PGAL. PGAL is nothing but your phosphoglyceraldehyde. In phosphoglyceraldehyde, to the third carbon, phosphate group will be attached. So we call it 3-glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. Okay, so from this point onwards, we have 2 molecules of glyceraldehyde. 6th step is the most important step. In the 6th step, what happens? Oxidation will take place. Glyceraldehyde is oxidized to acid. Glyceraldehyde 3-phosphate will become phosphoglyceric acid. Not just phosphoglyceric acid, it will become 1,3 bisphosphoglyceric acid. So, where did this extra phosphate come from? It came from phosphoric acid. Phosphoric acid will donate a phosphate group to phosphorylate this particular molecule. In the meantime, simultaneously, as aldehyde is getting oxidized to acid, simultaneously NAD plus will get reduced to NADH plus H plus. Okay, so that is the importance of this 6th step. In the next step, you have 1,3 bisphosphoglyceric acid, which will form 3-phosphoglyceric acid. That extra phosphate from the first carbon is donated to ADP and it will form an ATP. In the next step, 3-phosphoglyceric acid gets converted to 2-phosphoglyceric acid. Over there, there is no change in the number of carbons, no change in the number of phosphate. There are 3 carbons, 1 phosphate. Then 2-phosphoglyceric acid is converted to phosphoenol pyruvate. 2-phosphoglyceric acid to phosphoenol pyruvate, 3 carbons, 1 phosphate. In this, there is no change in number of carbons of phosphate, but a molecule of water will get released. And finally, in the last step, phosphoenol pyruvate gets converted to pyruvic acid. This had 1 phosphate, this has no phosphate. Extra phosphate is used in the formation of ATP. Is that clear? Why partial oxidation? Sagar, very good question. Unless glucose is completely broken down and carbon dioxide is released, it is not completely oxidized. So if you look at this 10-step process right here, is there any place where carbon dioxide was released? It is not, the complete energy in glucose in that carbon skeleton is not used up. We still have 3 carbon compounds. That still has energy. Once that is broken down, carbon dioxide is released, that gets completely oxidized. So far, complete oxidation has not happened. Hi Chandrakala, good morning. Nan super idini vegidira. Yeah, Himanshi will get to that. Is it clear so far? You should remember exactly what happens in each step here. Okay? Now let's see in which step what happened. I've added a slide in your notes so that you will get to know clearly which step what is taking place. All of the things that are happening, all of the events that are happening in glycolysis, you can know from this table. First step, what happens? Glucose to glucose 6-phosphate, ATP is utilized. Next, fructose 6-phosphate to fructose 1,6 bisphosphate, ATP is utilized. Then what happens? When there is oxidation, when phosphoglyceraldehyde is getting converted to 1,3 bisphosphoglyceric acid, NADH will get produced. You can think of, everybody knows ATP is the energy currency. It can be used directly in a cell to perform anabolic reactions. But NADH also has a lot of energy. You can think of it as ATP is like the cash currency, and NADPH, or I'm not NADPH, NADPH is in photosynthesis, NADH is in respiration. You can think of it like a check. If I'm buying like 20 rupees, if I'm buying medicine chilies on a street vendor, if I give him a check for 20 rupees, he'll be like, is this lady mad? Right? So if I have to get the money from the check, I have to go to the bank. Very similarly, NADH also has energy, but that cannot directly be used like ATP. NADH has to go to a bank, and that bank is present in the mitochondrial inner membrane. How exactly that will get encashed, we'll learn about later. For now, just remember, similar to ATP, NADH also has a lot of energy. In fact, every NADH can give us 3 ATP. So it has more energy compared to 1 ATP. Such an energy-rich molecule is produced during this oxidation step. Okay. So here that is going to get produced. If you remember, I told you there are 2 molecules here. So if there are 2 molecules of this, there will be 2 molecules of NADH and H plus produced, right? So in that step, NADH and H plus is synthesized. In the next step, you have 1,3 bisphosphoglyceric acid getting converted to 3-phosphoglyceric acid. Over there, 1 ATP is synthesized per molecule of here. So basically, there will be 2 ATP synthesized because there are 2 molecules. In the next step, we have phosphoglyceric acid getting converted to phosphoenol pyruvate. Here, 2 molecules of water is released. In the last step, 2 ATPs are synthesized. Just give me a minute. I'll drink a glass of water and come. Yeah. So it's like you have all of these, what do you call them? In banks, they give you all of these offers like you pay us 2,000 rupees right now. Then after a couple of years, we'll give you 4,000 rupees, like doubling of offers you will have, right? All these money schemes, very similar to that. In glycolysis also, in the first 5 steps, you can see how in 2 steps, ATP is used up. ATP is the currency is getting used. But after that, you can see how in 2 places, ATP will get synthesized. 2 ATPs will get synthesized in each step. So totally at the end of glycolysis, we have used 2 ATP, gained 4 ATP. So the gross ATP gained is 4, but 2 in that we've already used. So the net gain of ATP is 2. Simultaneously, 2 molecules of NADH and H plus is also gained. So what do we get at the end of glycolysis? We get 2 ATP and 2 NADH and H plus. Is that clear? So yeah, phosphorylation basically is the production of ATP, right? So ADP plus phosphate will form ATP, that is phosphorylation. You will learn about 3 different types of phosphorylation. One is photophosphorylation, one is substrate level phosphorylation, one is oxidative phosphorylation. What we saw right here and here, when one substrate is getting converted to another, ATP is getting synthesized. This type of phosphorylation is called substrate level phosphorylation. In photosynthesis also, you've seen in the light dependent phase of photosynthesis, ATP will get synthesized, right? So there also ATP is getting synthesized, phosphorylation is happening, but over there we call it photophosphorylation. Whenever NADH and FADH2, remember I told you that NADH is equivalent to 3 ATPs. For that it has to go to the bank, right? And where is that bank located? That bank is located in the inner membrane of the mitochondria. So if it goes to the bank and gets it encashed, at that moment, because of the electron transport system, because of the oxidation reduction reactions happening in the electron transport system, whatever ATP gets formed, that is called oxidative phosphorylation. So there are three things: photophosphorylation, substrate level phosphorylation, oxidative phosphorylation. Photophosphorylation you've learnt in photosynthesis. Substrate level phosphorylation we've learned right now in glycolysis. We'll learn about it in Krebs cycle as well. Oxidative phosphorylation is in what do you call it? ETS. Okay, is it clear? Now there are questions like most of you are asking. What is reverse glycolysis? What is repeat? Okay, Sneha says it's clear. Alright, let's move on. Let's solve questions based on concepts we've discussed up until this point. Okay, for those of you who are new to this session, I'll tell you how the poll works. For every question, I'm going to turn on the poll and give you like 30 seconds or 40 seconds, depending upon the difficulty level of the question. All you have to do is register or enroll on the Unity thing. You'll have a link at the pinned at the top of the chat box, and after that, once I start displaying questions, read it very carefully like it is your NEET exam and you're answering it in your NEET exam, and just type in the options in the comment box. Alright, so if everyone's ready, let's get started with the poll. Here's the first question of the day. Ta-da-da-da. Chandru, please focus on the class. Hi, hello, and socializing. All of that is not allowed in my class. ATP acts as the energy currency of the cell. Does it? Yes, it does. Gaseous exchange in plants takes place through stomata and lenticels. Yes, right. Plant parts like roots, stems, and leaves, respiratory rates far lower than animals. That is also true. In plants, usually proteins are oxidized to release energy. So the correct answer is option D. Who's going to tell me why it is wrong? 91% of you all have answered correctly. Abhilash. What could your name be? You answered the quickest, under 16 seconds, just around 15 seconds. Rohit, Shrisham, Saumya, Jatin, Anjali, Sahil, Sagar, Akash, Srijana, Naga, all of you have answered correctly. Who's going to tell me why option D is incorrect? Why is it incorrect? Yes, the most preferred substrate is not proteins, it is carbohydrates. I will write it here just so you know, if you're going through this notes later, you'll get the answer why this is wrong. Okay, now let's take a look at our next question for the day. All living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown is through Krebs cycle, AMP pathway, electron transport system, tricarboxylic acid cycle. Most of you are saying B. Nani or saying B, but try to type it only once. I'm not sure if you type it multiple times if the poll will register it or not. Okay, so you all are right, 100%, very happy. Yes, brilliant. Rohit is on top of the list. He's answered under 10 seconds. Brilliant, very impressive. Then we have Abhi, and then Anjali, Akash, Shri Sham, Sagar, Soumya, Naga, Jatin, Lord Ram, blessings, all of you all are there. At the end of the session, we'll see if this works. Does it work? Did you get an invite for the video call? No? Does it not work? Okay. Okay. So EMP pathway is the alternate name for glycolysis. This itself is the correct answer. Now let's take a look at our next question. Dash is a dash that acts as the energy currency of the cell. I'm sure everybody would know it is ATP, so there's not much confusion here. If only this option was ATP and nucleoside, then there might have been a confusion, right? ATP is a nucleotide. It is a nucleoside triphosphate basically. So the correct answer to this question is option B. It was very simple. I think I'll get 100% correct answers. Yes, a B is on top of the list again because he's answered in the shortest time. Yeah. Next, where does glycolysis take place? What is the site of glycolysis? Nucleoplasm, peroxisome, glyoxysome, cytoplasm. Where does it take place? Okay, everyone saying D. It does take place in the cytoplasm in all living cells, right? Still some of you have answered incorrectly. What was the confusion? What do you want me to try, Rohit? The video call thing? Are you getting a notification for video call? I don't know if it works. Every time I've tried, it doesn't connect. Okay. So the correct answer is option D. Okay, Saksham is in a hurry. Let's take a look at the next question. Select the correct option with respect to glycolysis. The first half of this pathway activates glucose for its breakdown. It's the only process of respiration in anaerobic organisms. It takes place in the matrix of the mitochondria. Option D, both 1 and 2. Yes, the first half is activation phase, it's also called, where ATP gets used up. Option B, it's the only process of respiration in anaerobic organisms. That is also correct. I had in fact written the exact same statement from NCERT on this slide. It takes place in the matrix of mitochondria. No, it does not take place in the matrix of mitochondria. It takes place in the cytoplasm. Both 1 and 2 are correct. So the correct answer to this question is option D. 84% of you all have answered. So Hale is on top of the list for this question. Everybody loves the things. Conversion of glucose to glucose 6-phosphate is catalyzed by which enzyme? I don't know what your name is. You're asking how you can try in the poll. You just have to type the correct answer in the comment box. No, Upadhyay, I'm not. Okay, hexokinase is the correct answer. Many of you have answered correctly. I think this will be a 100% no. What did you get confused with? Which other option? A, who is saying A? Prithvi Kumar is saying A. And Sushant says C. Otherwise, everyone has answered as B. Okay. We haven't even discussed the other enzymes. Some coaching classes teach them, but it's not in NCERT, so it's not important. Here's our next question. Which of the following reactions of glycolysis a molecule of water is removed? In which step exactly a molecule of water is removed? This is the last but one penultimate step, which is that you have to recollect. I'm getting mixed response. A, C, G. What is G, Upadhyay? C, A. Rohit, how can it be C and A? The correct answer is option A. Let me show it to you. Right here. Phosphoglycerate to phosphoenol pyruvate. This is the step in which water is removed. Right? So the correct answer is option A. Naga has answered correctly. Only 73% have answered. See, when you're asked these kind of questions, you should know very well the exact steps. Otherwise, you will lose marks. Okay? Tufani, whatever you think, that is true. If you think you can do it, that is true. If you think you can't, that is also true. It all depends on what you think about yourself in your mind. Okay? Lord Ram blessing. Who are you asking? Sheikh, good morning. Here's our next question. How many redox equivalents are removed from 2 molecules of 3-PGAL? Read the question carefully. Read the question carefully, Abhi. Read the question carefully, everyone. How many redox equivalents are removed from 2 molecules of 3-PGAL? Abhi has changed his answer now. Sagar says B. Jatin says B. I'm not going to be there standing behind you in your NEET exam and emphasizing on the word. Right? You should learn to read the question properly. So there's going to be 4. Per PGAL, it's 2. Right? If there are 2 NADH and H plus forming, then obviously there will be 4 redox equivalents. So the correct answer is option B. Any doubts in this question? Just 5% of you all have answered correctly. And that is Tufani, whatever is your name. Congratulations. I don't know if you answered by mistake or you understood the concept. Is there any doubt in this question? Does anyone have any doubt? Amrit, D or B? Make up your mind. Okay. So B is the correct answer. Let's take a look at our next question. Read the following statements with respect to glycolysis and select the correct ones. Does complete oxidation of pyruvic acid take place? Does it occur in the cytoplasm? Is it the only process in anaerobic organisms? Is there partial reduction of glucose? Does it produce ATP and NADH and H+? Is it complete oxidation of pyruvic acid? No. Does it occur in the cytoplasm? Yes. In anaerobic organisms, it is the only process, yes. Partial reduction of glucose, no. It is partial oxidation of glucose. It does produce ATP and NADH plus H+. So, B, C, E are correct. The correct answer is option B. Less than 50% of you all have answered correctly. Which question, which point was confusing? Who can tell me? What confused you? Partial reduction. Where did you get confused? Vijay Saxena, if you want to attempt the poll, participate in the poll, there must be a link, Unity link in the description box and at the top of the chat box, you have to enroll there. That's just like a 5-second process, but you have to enroll there. Otherwise, it will not, it will not register. D option, C, D and E. See, B is not wrong, right? It occurs in the cytoplasm. During glycolysis, there's partial oxidation of glucose happening, not partial reduction. Sheikh Sohail, good afternoon. Focus on the lecture. Okay, so that's about the questions regarding the introduction part and glycolysis. Is it clear so far? Has everyone understood? At the end of glycolysis, we have 2 molecules of pyruvic acid. Now, For 2 molecules of pyruvic acid, what is the fate? What is the metabolic fate of pyruvic acid? What is going to happen to that pyruvic acid molecule? Okay. One of 3 things can happen. It can undergo alcoholic fermentation, or it can undergo lactic acid fermentation, or it can undergo aerobic respiration. Fermentation itself, you know. If it is alcoholic fermentation or lactic acid fermentation, oxygen is not involved, so it is anaerobic. There is no oxygen. I'll write it like this otherwise. No oxygen involved. Okay. So alcoholic fermentation, lactic acid fermentation, both of them are anaerobic respiration. Aerobic respiration will utilize oxygen. First, let's study about anaerobic respiration, that is fermentation, and then move on to aerobic. Alcoholic fermentation. You've learnt about alcoholic fermentation, right? Soham, which question are you talking about? Jatin is asking, what is the net ATP gain and total ATP gain difference? See, you know, you may have heard your parents talk about gross salary and net salary, right? Gross is the total amount of salary that I will get. In how much ever I'm earning, a part of it will be my income tax that I will not get in my account. Minus the income tax, whatever, minus all the different types of tax, whatever salary I will get, that is the net salary. Very similarly, even during glycolysis, there are some ATP synthesis, there is some ATP utilization. Totally, how much ever ATP are synthesized, that is your net ATP gain. There will be some ATP used up, utilized also. Total is your gross ATP gain. From that gross, how much ever is utilized, if you minus what you get as a result, that will be your net ATP gain. So totally there are 4 ATP molecules formed in the second half of the cycle. That is your gross ATP gain. In the first 5 steps, 2 times ATP is utilized. So 4 minus 2 is 2, that is your net ATP gain. Sagar Mandal is talking about net primary productivity, gross primary productivity and all of that. Hexokinase is an enzyme. Schedule आपको क्या बोलते हैं उसे, कम्युनिटी पोस्ट में मिल जाएगा. Ethanol is ethyl alcohol. Okay, let's move on. In yeast, you know, yeast is also called brewer's yeast because it's used in the brewing industry for the synthesis of alcoholic beverages, right? Yeast under anaerobic conditions will respire anaerobically and produce alcohol as a result as one of the products. That's why it's called alcoholic fermentation. So what happens to pyruvic acid here is, in alcoholic fermentation, pyruvic acid, which contains 3 carbons, first undergoes decarboxylation. 1 carbon from pyruvic acid is removed in the form of carbon dioxide. You know that carbon dioxide is CO2, it will have 1 carbon, right? So first step, pyruvic acid undergoes decarboxylation to form acetaldehyde. Pyruvic acid to acetaldehyde. This is the first step. In this first step, an enzyme is utilized, and that is our pyruvate decarboxylase. Very clearly, as the name itself indicates, over here decarboxylation will take place of pyruvic acid. That's why this enzyme is called pyruvate decarboxylase. As a result, a 2-carbon acetaldehyde is formed. Okay, this acetaldehyde will undergo reduction to alcohol. Aldehyde to alcohol is reduction, right? So aldehyde will undergo reduction to form ethyl alcohol, and for that there is requirement of an enzyme called alcohol dehydrogenase. So over here reduction is happening. As reduction is happening here, remember NADH was produced in glycolysis, right? NADH will get reoxidized to form NAD+. NADH plus H plus will form NAD+. Is this clear? So overall, from alcoholic fermentation, how many ATP did we get? From glycolysis, we got 2 ATP, 2 NADH and 2 pyruvic acid, right? So if it is going through alcoholic fermentation, whatever 2 ATP we got, that is all the ATP the cell will get. whatever NADH was reduced, whatever NADH was formed as a result of reduction in glycolysis, that will get reoxidized over here. So NADH was supposed to have 3 ATPs, but if it's going through alcoholic fermentation, those 3 ATP in NADH the cell will not get, because in this step that NADH is getting reoxidized into NAD plus H plus. Okay? So, is this clear? If a cell undergoes alcoholic fermentation totally from the entire process, glycolysis plus alcoholic fermentation, everything included, a total of 2 ATP is all the cell will get. Okay? Now let's talk about lactic acid fermentation. In the chapter Microbes in Human Welfare, we've also learnt about lactic acid bacteria which are involved in the production of curd, right? So they are called lactic acid bacteria. They produce lactic acid by breaking down certain components in a cell. So when does this lactic acid get produced? In some bacteria. And also you may have noticed how when you're exercising very severely, and severely is not the right term, very rigorously, and you're not breathing properly, your body is not getting enough oxygen. But your muscles are contracting, relaxing, which requires a lot of energy. So if energy is required and you're not breathing oxygen properly, you're not taking in enough air into your body, then your muscle cells will not get sufficient oxygen. They do not get sufficient oxygen, but they have to undergo respiration to produce ATP. Then the alternate route or the alternate path that it takes is lactic acid fermentation. In lactic acid fermentation, the pyruvic acid gets converted into lactic acid. The difference between alcoholic fermentation and lactic acid fermentation is pyruvic acid has 3 carbons, ethyl alcohol has two carbons. During alcoholic fermentation, 1 carbon dioxide gets released. But in lactic acid fermentation, that is not the case. Pyruvic acid also has 3 carbons, lactic acid also has 3 carbons. So this conversion will happen in the muscle cells when there is not sufficient oxygen. Okay, so that is why people say you get cramps also because of the accumulation of this lactic acid. Okay, the enzyme that is required for the conversion of pyruvic acid into lactic acid is lactate dehydrogenase. Even over here, NADH will get reoxidized. So even over here, you will get a total of 2 ATP only. Only what was produced in glycolysis, that is all the ATP that will that the cell will get. Okay, so this happens in bacteria and it also happens in our muscle cells when there is not sufficient oxygen. I hope it is clear. Hi Sakshi, it's okay, better late than ever, never. Is it clear so far? Okay. So fermentation is anaerobic and aerobic respiration makes use of oxygen, fermentation does not. Why have most organisms evolved to undergo aerobic respiration and not anaerobic respiration? Because anaerobic respiration or fermentation, there are a few drawbacks. What are the drawbacks? The first drawback is that glucose has some amount of energy. If it's undergoing anaerobic respiration, glycolysis only is happening. So all of the energy in glucose is not broken down. Glucose is not completely oxidized, only partially it is oxidized. If this partial oxidation is happening, energy, not much energy is released. We know exactly only 2 ATPs are released. In contrast, in aerobic respiration, ideally we'll get 38 ATP. Look at the difference, 38 and 2. Right. So in anaerobic respiration, the amount of energy that is released is very, very less. Also, whatever energy is present in glucose, remember I told you when glucose gets broken down, all of the energy will not be trapped in ATP molecules, even in aerobic respiration also. Some amount will obviously be lost as heat, even if complete oxidation happens. But over here, only partial oxidation is happening. Less than 7% of energy in glucose is released, and of that even less is trapped in the form of ATP. Okay, less than 7% of energy is released, and even in that a very small amount is trapped in the bonds of ATP. First most important point is we do not get much energy by performing anaerobic respiration. That is why. What is the second step? What are the molecules we are getting as products of anaerobic respiration? One is an alcohol, one is an acid. Excess of both is not good for our cells, right? Both of them can turn toxic at high concentration. Both of these are hazardous. Both acid and alcohol at high quantities are not good for our cells. That is the other reason why most organisms, unless absolutely required, do not undergo anaerobic respiration. Okay, correct, Upadhyay. Shwetank. Shwetank, is that the correct pronunciation of your name? Yes, you get 2 ATPs, Rohit. Okay, so there are a couple of questions asked in NCERT for which you should know answers. First question is poison the, so basically you know that in large fermenters they grow yeast and they give it some nutrient material, nutrient medium, some sort of broth, and they will grow the yeast in that in order to produce alcohol, right? So whenever in the medium the alcohol concentration reaches greater than 13% or about 13%, this is an important number for you to remember. This also is an important number for you to remember. Okay, when it's about 13%, the yeast will die. It will start dying. All right, but there are certain alcoholic drinks in which the concentration of alcohol or the content of alcohol is much more than 13%. If you consider like whiskey and rum and many other different types of alcoholic drinks, the alcohol content in that exceeds 13%. Okay, how is that possible? That is one question. The second question is, if the alcoholic drink is naturally fermented, what is the maximum concentration of alcohol present in it? If it is naturally fermented and if yeasts are dying at 13% maximum concentration, then it should be 13% or less than that. Equal to 13% or less than that. But we do see alcoholic beverages that have content of alcohol in it more than 13%. How do we get it? We get it because of distillation. You may have learnt about fractional distillation with respect to petroleum in your lower grades, right? Based on the boiling point, it will evaporate and you can collect it. So what happens in fractional distillation is whatever diluting substances are there, water and all of those are there, they will be removed such that the alcohol concentration becomes very high. So that is how beverages are produced that have high alcohol content like whiskey and rum. Okay. Yes, in microbes chapter you will learn about it. क्या repeat करना है साक्षी? Toddy about that you learn in microbes. Even toddy has very little alcohol content. Okay. Yeah, we have some questions based on fermentation. Is everybody ready? 3, 2, 1, let's get started with the questions. Here is our first question based on fermentation. Yeast poison themselves to death when the concentration of dash reaches about 13%. Very simple question. Why did I even add this? Let's see if there are some people who will get this also incorrectly. Everyone saying C. The correct answer indeed is C. 100% correct. Venkat Pushpa has answered in around 13 seconds. Naga has answered in 14 seconds. Very good. Oh, on top 3 we have 2 girls. Top 4 is also a girl. Amala, Elizabeth. Nice to see both boys and girls participating. Generally it's dominated by boys. The top 3 ranks. Next question, read it yourself. Two options can get confusing at this point. Let's see how many will answer correctly. Akash says A, the rest of you all are saying C. And the correct answer is C. Okay, everyone's answered correctly. Lactic acid dehydrogenase is the correct answer. Decarboxylase is wrong. Whenever pyruvic acid is getting converted to lactic acid, decarboxylation is not happening, right? Both of them have 3 carbons each. So, correct answer is option C. Naga, Soumya, Anjali at the top 3 spots. Next question. In muscles, during exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to. Mad hitter always says CCC, BBB 3 times, like order, order, order. Okay, now he said it 5 times. Shrisham says B, everyone says C. The correct answer, who is saying D? The correct answer is lactic acid. Because of the accumulation of lactic acid is the reason why you feel cramps. Abhi is on top of the list. He's answered in under 6 seconds. Very impressive. Just around 7 seconds. Shwetank also. Anjali, all of them have answered in under 10 seconds. It was a very simple direct question. Here's our next question. Net gain of ATP molecules for each molecule of glucose in fermentation. How many ATP do we get from fermentation? 8, 10, 4, 2. There's confusion between D and C. Okay. D is the correct answer. We get only 2. Here's our next question. It's a statement based question. Read carefully without making any mistakes. You have to identify the correct statement with respect to fermentation. It is a metabolic process in which oxidation of substrate occurs without any external electron acceptor. It is a reaction occurs in the cytoplasm of aerobic bacteria and the what is the correct statement? The correct statement is A. Adil says A. Rohit, where is your answer? Pushpendu Karan is saying B. Sakshi, my name is Shalini. Anu also says A. Yeah, so A is the correct answer. Let's see why the others are not. I expected most of you to answer this incorrectly. Naga, Sagar, Sarika, just the 3 of you. Okay, have answered correctly. It is a metabolic process in which oxidation is happening, but there's no external electron acceptor. That is correct. It is a reaction occurring in the cytoplasm of aerobic bacteria and in mitochondria of eukaryotes. No, even in eukaryotes also, it happens in the cytoplasm itself. It involves synthesis of one molecule of ATP and NADH. NADH is not even synthesized. It gets synthesized, it gets used up. So it's not even counted. We get 2 molecules of ATP, not 1. It involves complete oxidation of pyruvate, not at all. It is incomplete oxidation of glucose, not pyruvate. Right. So the correct answer is option A. Now, here is our next statement based question. Read carefully, don't make any mistakes. How many of the above statements are right? Above statements, shouldn't it be below statements? Lactic acid fermentation releases carbon dioxide only. Does it release carbon dioxide at all? It doesn't, right? Alcoholic fermentation is commonly seen in yeast and certain bacteria. Yes. Alcoholic fermentation in yeast, certain bacteria you see lactic acid fermentation. One of the crucial events in aerobic respiration is the complete oxidation of pyruvate. Pyruvate, yes, does get completely oxidized. The maximum concentration of alcohol in naturally fermented beverages is not 7%, right? It can go up to 13%. So, what was your answers? Only one question, one statement is correct. That is statement C. So the correct answer is option D. Wow, brilliant. What were your answers? Which other statement did you think is correct? What were you saying? Two statements. Which other statement did you think is correct? In certain bacteria, you see lactic acid fermentation, no? A is not correct. In lactic acid fermentation, carbon dioxide is not even released. In yeast, you see alcoholic fermentation. In bacteria, you see lactic acid fermentation. Is it clear now? According to NCRT. Yeah, silly. So read carefully. Here's our next question. Read the given statements and choose. Okay, I will not read it. You read it yourself. Just a moment. I will change the option. There is a mistake. Let's make this only B is correct because it was repeated. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. So this is incorrect. During glucose activation phase, that is the first 5 steps, first half of glycolysis, 2 ATP are consumed. Yes, 2 ATP are consumed. This is correct. So only option B is correct. The correct answer is option B. Sujata has answered in less than 2 seconds. Sujata, where are you? Where were you all this while? Naga is doing really well today. Sagar also, Abhi also. All of you all are doing great. Nani, Akash, Elizabeth, Saumya, Amreen, Fatima, all of you have answered this correctly. Somebody said I'm cheating. Who is saying I'm cheating? Okay, I'm so sorry. I did not read the question properly. Venkata Pushpa caught me. I'm so sorry. But still, how can it be? This has to be B, otherwise it won't work, right? I read it incorrectly. I thought this is only B is correct. You will get +4, I get -1. B is not incorrect, B is correct. Clear now? I had read it wrong. My bad. Here is our next question. With respect to the above figure, which of the following statements is correct? This is the figure we are talking about. Okay. Read it properly. Sakshi, I can speak Hindi. Okay, decently manageable, but not very great. Shwetank says A, everyone else says B, Sagar is saying C, Pushpendu also says C. Who said alcoholic fermentation is carried out only by prokaryotes? Yeast is a prokaryote or eukaryote? What is this trying to show? The first reaction is your lactic acid fermentation. This reaction is your alcoholic fermentation, right? What does first option read? The figure shows the fate of pyruvic acid in both aerobic as well as anaerobic pathway. Not at all, both of them are anaerobic, so that is incorrect. X to Y or A to B both represent reoxidation of NADH to NAD plus. Yes, that is correct. Second one is alcoholic fermentation. It's carried out by prokaryotes only. No, it's not carried out by prokaryotes only. For example, we have yeast, which is a eukaryote, right? D. This should be one. Is oxygen produced as a byproduct during the process? Oxygen is not produced so far. So that is incorrect. So what is correct over here is option B. Is it clear, everyone? Hitesh has not understood. Shwetank understood. Anybody else has doubt? Why is Shrisham saying C? Hi Arjun, namaskara, hegidira? Hitesh, have you understood now? See, pyruvic acid to ethyl alcohol. I will show you the slides. Wait. Instead of writing. Even during alcoholic fermentation, pyruvic acid to ethanol, reoxidation of NADH will happen. During conversion of pyruvic acid to lactic acid during lactic acid fermentation, reoxidation of NADH will happen. Right? So that is what is shown here. They have not given NADH to NADH plus H plus to NAD plus. They have just mentioned it as A and B. So here they are saying both X to Y in alcoholic fermentation and A to B in lactic acid fermentation, both of them represent reoxidation of NADH to NAD plus. Also, I missed one thing. Here D is A here is this one. A is carbon dioxide, not oxygen. Carbon dioxide is produced as a byproduct, not oxygen. That makes this also incorrect. So the correct answer here is option B. Hitesh, why do you think C is correct? I told you clearly. Alcoholic fermentation takes place in eukaryotes also. Yeast is a eukaryotic organism. That is the example. Okay. Here's another similar question. What does A, B and C depict in the given pathway of anaerobic respiration? Where is A, B, C? Find that out first. A is here, B is here, and where are you? C? C is here. Majority of you are saying B, some of you are saying D. The correct answer is option B. Okay, first I'll explain, then we'll see. Here you have glucose. They are showing multiple arrows here. That means it is a multiple steps. After many steps, you get A. When A is getting converted to 3-phosphoglyceric acid, NADH production is happening. Look at the way the arrow is pointed. NAD plus to NADH. NAD plus is getting reduced to NADH. Simultaneously, if you remember, an aldehyde gets reduced, oxidized to an acid. Which is that aldehyde? It is our glyceraldehyde 3-phosphate. So this is PGAL. Right? Correct. And here you have pyruvic acid. Pyruvic acid is formed from what? Phosphoenol pyruvate will give rise to pyruvic acid. So B is phosphoenol pyruvate. Pyruvic acid has 3 pathways. Either it can undergo alcoholic fermentation or lactic acid fermentation or aerobic respiration. Over here they have shown Utilization of NADH or reoxidization of NADH. That means both of these are fermentation. On one end, they've already given ethanol plus carbon dioxide. So this is alcoholic fermentation. On the other side, carbon dioxide is not released. It should be forming a 3-carbon compound. Which is that 3-carbon compound? It is lactic acid. So C is lactic acid. There is a mistake here. Pyruvate, there is a typing mistake. This should be phosphoenol pyruvate. It has gone here. Okay, this is lactic acid. This is phosphoenol pyruvate. Is it clear now? Is it clear? Has everyone understood? Is there any confusion? Okay. Anjali didn't understand? So B is the correct answer. 3333. 33.33 is the number of people who've answered correctly. Elizabeth has answered the quickest, less than 25 seconds. Congratulations. Then we have Vishal and Harika as always. Okay, so that's about anaerobic respiration. We've solved a decent number of questions. Hopefully, you have understood all the concepts. किसने बोला है इंडिया में हिंदी एक ही लैंग्वेज है? इंडिया में बहुत सारे ऑफिशियली रिकॉग्नाइज्ड लैंग्वेजेस है, तो इंडिया हिंदी इज नॉट द नेशनल लैंग्वेज ऑफ इंडिया. Okay, let's take a look at the leaderboard. Osimum is really curious to see if he's on top of the list. Okay, Akash is on top of the list. 14 out of 17 questions he's attempted is correct. Sagar, 13 out of 16. Abhi also has attempted 13. Look at the difference in the time. Both of them have attempted the same number of questions and answered the same number of questions correctly. But the reason Sagar is on top is because of that 2-second, almost just 2-second difference. Okay. Then we have Naga, who has been answering. His average time is very less. He's answering in less than 20 seconds approximately. Out of the 14 questions he's attempted, only 12 he has answered correctly. Why is it that you have attempted only 14 questions? I need to understand. Happens sometimes, Rohit, but you keep a track of it yourself. If it's not getting reflected here, you keep a note of how many you're getting correctly, why you're getting incorrectly, all that you have to track. Okay? So, yeah. Then we have Jatin, Anjali, Soumya, Venkat Pushpa, Rohit and Srijana in the top 10. Now let's move on and talk about aerobic respiration. Okay, so you have a network problem. But even if you're not answering here, because of some reason you're not able to answer, you keep a track of it on the side. Okay? Soumya, what is the reason you lost? Lost or last? It's okay. At least you know where you're answering incorrectly, so you can study that better. Right? So all this is like a test for you to find out which concepts you have not understood properly. Sheikh, please stop spamming. Thank you if you think I'm intelligent. So next we have aerobic respiration. Obviously, if it's aerobic respiration, it will require oxygen, right? So aerobic respiration in eukaryotes will take place inside mitochondria. That is the site of the next steps of aerobic respiration. Yeah, if aerobic respiration is taking place, then Arjun, yes, I'm doing good. Yes, Sagar, that is exactly the point of this practice session. You should find out where you're making mistakes and learn that better. Yeah, so if aerobic respiration is taking place, there's going to be a complete oxidation of organic substances, and this happens in the presence of oxygen. And in this process, carbon dioxide is going to get released. Along with carbon dioxide, water and a large amount of energy in the form of ATP is going to get released. Okay, we see this in most higher organisms. So, what happens to the pyruvate that was formed in the cytoplasm? Remember, glycolysis took place in the cytoplasm. Pyruvic acid was formed in the cytoplasm, but we are saying aerobic respiration takes place inside the mitochondria. So the first thing that needs to happen is the pyruvic acid that's produced in your cytoplasm has to be transported into the mitochondria. So that is exactly what happens. Pyruvate will get transported from the cytoplasm into the mitochondria. So in aerobic respiration, there are two very important events. What are those? Let's find out. First of all, I've been emphasizing since the beginning that if it is aerobic respiration, complete oxidation is going to happen. When complete oxidation is going to happen, maximum amount of energy is trapped in the form of ATP. If complete oxidation is happening, it's going to happen in a stepwise manner. When complete oxidation is happening, a lot of energy is released in the form of ATP, right? So when complete oxidation is happening, of pyruvate. Right now we have pyruvate. Glucose is partially oxidized and has given us two pyruvic acid molecules. Now that will continue to get oxidized. Okay. So when that gets oxidized, hydrogen is removed simultaneously. Remember, let's take a look at this initial equation that will help you understand. Where did it go? I can't find it. Never mind. Yeah. So what happens is in pyruvic acid, hydrogen, whatever hydrogen will be present, that hydrogen will be removed. As pyruvic acid is getting oxidized, hydrogen will be removed and that hydrogen is used in the formation of energy rich molecules like NADH. Okay. And carbon dioxide also will be released. If you talk about the respiration equation, we say that glucose plus oxygen will give us energy plus water plus carbon dioxide, right? So whatever carbon, oxygen, hydrogen is there in pyruvic acid, carbon and oxygen will get released as carbon dioxide, hydrogen is removed and NADH formation will take place. This NADH will later go to the bank and get it encashed and form ATP. Alright. So the first thing is whatever pyruvic acid in that, whatever carbon and oxygen are there, they are removed as carbon dioxide. Whatever hydrogen is there, that will be used in the formation of NADH molecule. Alright, so that's the first step. Second step, the passing on of the electrons removed as a part of the hydrogen to molecular oxygen while ATP will get simultaneously synthesized. Okay, so whatever NADH will form, from that the electrons will get transported through a series of electron carriers and ATP will get synthesized. That is your electron transport chain. Where does this oxidation happen? This will happen in your link reaction as well as in your Krebs cycle. Is it clear so far? Not everything that the Google search gives you is correct, Sujal. I think at your age you should have that much common sense. Yeah, I do agree. Vipin Sharma is the goat of botany. Kaushik, if that makes you feel better. Thanks, Ashutosh. So, is it clear? Till now, whatever I have told, is it clear? Moving forward, we are going to read about link reaction, Krebs cycle, etc. In link reaction and Krebs cycle, what is going to happen? Decarboxylation will happen, carbon dioxide will get removed, NADH will form. In ETC, what will happen? NADH, whatever electrons it has, it will donate that electron to the electron carriers in your electron transport system and simultaneously ATP will get synthesized. Okay? Is it clear? Okay, let's move on and talk about why is this repeated twice. I will delete it later because it will get repeated in your notes otherwise. Yeah. So let's talk about oxidative decarboxylation. Over here, oxidative decarboxylation is also called link reaction. Why do we call it oxidative decarboxylation? In this reaction, oxidation is happening and also decarboxylation is happening. Whenever decarboxylation happens, what happens? A molecule of carbon dioxide will get removed. Over here, this occurs in the mitochondrial matrix. Everybody knows the structure of mitochondria, right? So if this is your mitochondria, it has two membranes. Outer membrane is smooth, inner membrane has many enfoldings called cristae. And inside, this space that is present inside your mitochondria, that is called your mitochondrial matrix. So this is the location of your link reaction. Where did the cytoplasm, where did glycolysis happen? Glycolysis happens in the cytoplasm. So the pyruvic acid that is produced in the cytoplasm will be transported into the mitochondrial matrix, and within the mitochondrial matrix, this oxidative decarboxylation will take place, right? So what exactly happens during oxidative decarboxylation? So oxidative decarboxylation, oxidation will happen, decarboxylation also will happen. For this, there are certain coenzymes required. Which are those coenzymes? Co-A I have written here. This is nothing but coenzyme A. NAD plus is also a coenzyme. NAD, I have not given you the full form, right? NAD is nicotinamide adenine dinucleotide. What is NADH then? The reduced form of NAD is NADH. Okay. So pyruvic acid along with these coenzymes, coenzyme A and NAD plus, in the presence of magnesium ions catalyzed by the enzyme pyruvate dehydrogenase will form acetyl coenzyme A. We all know that pyruvic acid has 3 carbons. We all know during this reaction decarboxylation is happening. So, one carbon dioxide is released, removed from pyruvic acid, 1 carbon is gone. Acetyl coenzyme A is forming. So, how many carbons will acetyl coenzyme A have? It will have 2 carbons. Is it clear? So, pyruvic acid plus coenzyme A plus NAD plus in the presence of pyruvate dehydrogenase and magnesium ions, remember the names of enzymes and all this very well, will form acetyl coenzyme A, which is a 2 carbon compound. Along with that, a carbon dioxide is released. That is what we call decarboxylation. While pyruvic acid is getting oxidized to acetyl CoA, NAD plus will get reduced to NADH and H plus. So in this process, oxidation is happening, reduction is happening, decarboxylation is happening. What is getting oxidized? Pyruvic acid is getting oxidized to acetyl CoA. What is getting reduced? NAD plus is getting reduced to NADH and H plus. Decarboxylation is also happening because a 3 carbon compound is forming a 2 carbon compound releasing a carbon dioxide. That is why it is called oxidative decarboxylation. The alternate name for oxidative decarboxylation is your link reaction. Okay? Is it clear? Any doubts here? Okay, cool. Soumya has understood? Okay, cool. If you're trying to offend me by taking names of other teachers, know that I also look up to them. I think they are great teachers too. You cannot offend me. My threshold for, getting offended is here. You can't even reach there. All right. So I love your teachers as much as you do. Next, what happens to acetyl-CoA? Now, this acetyl coenzyme A will enter your Krebs cycle. It is a cyclic pathway. That's why it's called Krebs cycle. Why do we call it Krebs? Just like we call Calvin cycle, the C3 cycle, Calvin cycle, because he's the one who elucidated it. Just like we call glycolytic pathway, EMP pathway, because those 3 people elucidated it. And we call this pathway Krebs cycle because it was a person called Hans Krebs who was the first to elucidate the pathway. Okay, so this acetyl-CoA will now enter the cyclic pathway that is called Krebs cycle. It is also called citric acid cycle. It is also called tricarboxylic acid cycle. In short, TCA cycle. Why it is called so, I'll tell you in just a bit. Okay. Now, let's talk about Krebs cycle. Very important. Very similar to how you have so many steps in glycolysis. At different steps, different things are forming. Even over here, you have like 8 steps. You should remember it very, very well. Okay. Now, we are starting with acetyl-CoA. If you take a look at your Krebs cycle diagram or the schematic representation in your NCERT, it will not show this step. But I've added this one intermediate extra here because this is not given to you in your diagram, but it is mentioned in the text. Okay, therefore, I've added this one extra intermediate. If you're revising Krebs cycle, you can take this diagram because it has everything there is everything that is there in the text. Okay. Also in Krebs cycle, in your textbook, it shows the link reaction as well. How acetyl-CoA is getting, how pyruvic acid is getting converted into acetyl-CoA, that is also shown here, but that is not officially a part of the Krebs cycle. That is different. That's a link reaction. Oxidative decarboxylation, this is different. This is Krebs cycle. Krebs cycle begins with acetyl-CoA and oxaloacetic acid getting condensed. Okay. I also have a trick for Krebs cycle. I'll share it in just a bit. Now, the first step in Krebs cycle, remember the number of carbons here. It's important. Where all NADH is forming, where all decarboxylation is happening, where all FADH2 is forming, where substrate level phosphorylation will take place. All of that we have to remember. First step, we'll start from here. We'll go in the clockwise direction. We have acetyl-CoA as a product of the link reaction. Acetyl-CoA has 2 carbons. This 2-carbon compound acetyl-CoA along with oxaloacetic acid which has 4 carbons, there is condensation reaction. Wow. Condensation reaction that takes place where this 2-carbon acetyl-CoA will combine with this 4-carbon. Not this. Just give me a moment. Yeah. So this will combine with this 4-carbon citric acid, 4-carbon oxaloacetic acid to form a citric acid. 2-carbon, 4-carbon, 6-carbon. For the formation of citric acid here, an enzyme is required and the name of that enzyme is citrate synthase. Remember this enzyme. In this process, one molecule of coenzyme A is removed. Oxaloacetic acid 4-carbon, acetyl-CoA 2-carbon, condensation reaction, catalyzed by enzyme citrate synthase forms citric acid. Now you understand why we call this citric acid cycle. It's called citric acid cycle because the first stable product that is formed here is citric acid. Why do we call it tricarboxylic acid cycle or TCA cycle? This citric acid, which is the first product, has 3 COOH groups. It has 3 carboxylic acid groups. That's why it's called tri. Tri means 3, right? So that's why it's called tricarboxylic acid cycle. Okay? That is your first step. In the next step, citric acid gets converted into isocitrate. There is nothing major changes that are happening in this step. In the next step, isocitrate, see here there is no change in the number of carbons. Isocitrate will get oxidized and form a 5-carbon compound called alpha ketogluteric acid. Here there are 6 carbons, here there are 5 carbons. Where did that extra carbon go? It is liberated as carbon dioxide. Clear? It is liberated as carbon dioxide. I told you over here isocitric acid or isocitrate is getting oxidized to alpha ketoglutarate. Simultaneously, an NAD plus will get reduced to NADH. So if you look at it, this is very similar to what happened here. There was decarboxylation, 3-carbon compound lost a carbon dioxide, became a 2-carbon compound. Simultaneously, there was a synthesis of NADH. We call this oxidative decarboxylation. Something very similar is happening even here. This is also oxidative decarboxylation where isocitrate is getting oxidized to alpha ketoglutarate, carbon dioxide is getting lost, and also NADH is forming. Understood? Citrate synthase comes here. Shahil, you're back to this class. Why are you so late? Okay? Is it clear so far? Let's consider the next step. Alpha ketogluteric acid, which has 5 carbons, will further undergo decarboxylation and form a 4-carbon compound called succinyl coenzyme A. This is not mentioned in NCERT. I've added it extra because it's there in the text but not in the diagram. In this process, again, if you notice, alpha ketoglutarate is a 5-carbon compound, succinyl coenzyme A is a 4-carbon compound. Here decarboxylation is happening, 1 molecule of carbon dioxide is released. Here again, alpha ketoglutarate is getting oxidized to succinyl CoA. Simultaneously, NAD plus gets reduced to NADH and H plus. So this also is oxidative decarboxylation. Megha, I'll give you the mnemonics after I finish this cycle. Is it clear now? So far, you saw in 3 places carbon dioxide is released. Which are the 3 places where carbon dioxide is released? 1, 2 and 3. If we saw in the beginning of the lecture, we saw the equation of respiration. From 1 molecule of glucose, we said that 6 molecules of carbon dioxide will get released. Have 6 molecules of carbon dioxide gotten released yet? Have 6 molecules gotten released yet? Yes or no? Yes or no? See here, we saw over here 1, over here 2 and over here 3. 3 molecules have gotten released. But how many pyruvic acid have we taken? Per molecule of pyruvic acid, 3 molecules of carbon dioxide are released. But how many pyruvic acid do we have? We have 2 pyruvic acid. So if you consider 2 pyruvic acid, already 3 carbon dioxide molecules have released. Okay. So glucose is not even fully oxidized, but 3 molecules of carbon dioxide is already released. So far, water has not been produced, oxygen has not yet been used. So even in Krebs cycle or in link reaction, oxidation has not yet been used. We are saying that this is aerobic respiration. We know that aerobic respiration means there is involvement of oxygen. But though this is part of aerobic respiration in link reaction and in Krebs cycle, oxygen has not yet been utilized. The oxygen's role comes much later. Okay, keep that in mind. Up until this point, 6 molecules of carbon dioxide have already been released. Okay, so here alpha ketoglutarate in the next step will get converted into succinyl CoA where another carbon dioxide molecule will get released. Okay, now this succinyl coenzyme A, further there is no more decarboxylation, all are 4 carbon compounds. Okay, so now this succinyl coenzyme A will get converted to succinic acid. When succinyl coenzyme A gets converted to succinic acid, a molecule of GTP will get synthesized. Now what is GTP? GTP is very similar to ATP. You know, in DNA or in nucleic acids you have ATGC, right? Adenine, thymine, guanine, cytosine. So here in adenine, if adenine is there in the nucleotide, it is called ATP. If guanine is there in the nucleotide, it's called GTP. But the amount of energy present in ATP is equal to the amount of energy present in GTP. You can consider this as an equivalent for ATP. What happens immediately after this GTP gets formed is, once this GTP forms, it will split again to form GDP and inorganic phosphate. It will provide that inorganic phosphate to ADP and synthesize a molecule of ATP. Okay, so this is coupled. This is coupled to ATP synthesis. So in this step, ATP is forming. Who can tell me what type of phosphorylation this is? Is it photophosphorylation? Is it substrate level phosphorylation? Is it oxidative phosphorylation? What type of phosphorylation is taking place here? I'm telling you that an ATP is forming. Yes, over here substrate level phosphorylation is taking place. In photosynthesis, phosphorylation, in glycolysis and Krebs cycle, whatever ATP gets synthesized, that is substrate level phosphorylation. Whatever ATP we obtain from NADH and FADH2, that will be your oxidative phosphorylation. Okay, so here a substrate level phosphorylation is taking place. Per pyruvic acid, 1 ATP or GTP we are obtaining. What do we have here? Succinic acid. Succinic acid in the next step will get converted into fumaric acid. For succinic acid to get converted to fumaric acid, whenever it is getting oxidized, simultaneously a molecule of FADH2 is getting produced. What is FADH2? I think I've written it in the next slide. FAD is flavin adenine dinucleotide. NADH, like I already mentioned before, nicotinamide adenine dinucleotide. Okay, so here FADH2 is produced. FADH2 is similar to NADH. It can also provide ATP. One NADH will give us 3 ATP, one FADH2 will give us 2 ATP. That is how exactly it happens, we'll learn moving forward. Okay, so succinic acid in this step is getting converted to fumaric acid. Over here, I want you to remember one point very clearly. There is an enzyme called succinate dehydrogenase. This succinate dehydrogenase is involved in the oxidation of succinic acid to fumaric acid and simultaneous reduction of FAD plus to FADH2. Every step in this Krebs cycle is enzyme mediated. We are learning only the important enzymes. I told you about citrate synthase. After that, I have not given you any enzymes. But I am telling you importantly succinate dehydrogenase because we are going to come back to this later in ETS. Where is this Krebs cycle taking place? Krebs cycle is taking place in your mitochondrial matrix. It's taking place in the mitochondrial matrix. So whatever enzymes are required for Krebs cycle, they are located in the mitochondrial matrix itself, except for this one enzyme succinate dehydrogenase. This succinate dehydrogenase is present in the inner membrane of the mitochondria. All others are present in the matrix of the mitochondria. Succinate dehydrogenase is located in the inner mitochondrial membrane. This will act like a link between your Krebs cycle in the matrix and the ETS in the inner mitochondrial membrane. Okay, so that is why it's important. Remember this. In the next step, fumaric acid gets converted into malic acid, which is also a 4-carbon compound. Last step, malic acid to oxaloacetic acid, another molecule of NADH is produced. Is it clear? Is it clear so far? Any doubts? I'll give you the mnemonic. Our city is kept secure and safe from molesters. Okay, different people have different ways of remembering this. It's all right. Here is the mnemonic that I remember it. Our is for oxaloacetic acid. We are going to start from this point. Our active. It should be our active. Okay. So, our is oxaloacetic acid. Active acetyl-CoA is our active city is citric acid, is first two words isocitrate, kept keto, alpha ketoglutarate, safe, secure. Remember, here you have succinyl-CoA, here you have succinic acid. Both of them are starting with C. If you want to find out which is first, if you want to remember which is first, see, secure has a C in it. So, succinyl-CoA comes first. Our active city is kept secure and Safe, the other S, from fumaric acid, molesters malic acid. You can write it down. Doesn't matter. You can write whatever you want. हंसते जाओ अनमोल, हंसते जाओ. पेपर में मार्क्स भी ऐसे ही हंसेगा तुम्हें देखकर. You can change it to whatever you want. I have written molesters because malic has an L, molesters has an L. That way you can remember well. But you can change it and modify it to whatever works for you. Our active city is kept secure and safe from molesters. Here, what's important to remember is how many NADH is produced, in how many steps NADH got produced? In one. Yeah. So here in one step you have NADH, second step you have NADH, third step you have NADH. In 3 steps of Krebs cycle, NADH is produced. In 1 step of Krebs cycle, FADH2 is produced. In 1 step of Krebs cycle, GTP is produced. In 2 steps of Krebs cycle, carbon dioxide is released. Whenever there is conversion happening from 5 carbon to 4 carbon, whenever conversion is happening from 6 carbon to 5 carbon, carbon dioxide is released. Okay? So that's about your Krebs cycle. This is the overall equation of your link reaction and Krebs cycle combined. What are the different things that we saw? We started off with pyruvic acid, which has 3 carbons, right? This was the end product of glycolysis. So from pyruvic acid, you know that in the link reaction, 1 NADH is produced, and in the Krebs cycle, 3 NADH is produced. I'll write Kc. Krebs cycle, 3 NADH is produced. FAD plus, this is in the Krebs cycle. Water also, but we'll learn about it later. ADP in the Krebs cycle. Pyruvic acid, carbon dioxide, decarboxylation. 1 carbon dioxide is released in the link reaction. 2 carbon dioxide are released in the Krebs cycle. 4 NADH, 1 in your link reaction, 3 in your Krebs cycle. Yeah, so this is entirely in your Krebs cycle. This is also in Krebs cycle. Okay? So from link reaction and Krebs cycle combined, from 1 molecule of pyruvic acid, we will be getting 4 molecules of NADH, 1 molecule of FADH2, 1 molecule of ATP, and 3 molecules of carbon dioxide released. This is obtained. Is that clear? So whatever ATP is produced, ATP can be directly used as the energy currency. NADH and FADH2, like I told in the beginning, they cannot be directly used. That is, like the check, for encashment it has to go to the bank. So NADH and FADH2 will have to go to the bank, which is your electron transport system that is located in the inner membrane of the mitochondria. Link reaction and Krebs cycle, both of them took place in the mitochondrial matrix. Now electron transport system will take place in the inner mitochondrial membrane. So far, let's see how many ATP are produced in different steps. In glycolysis, net gain of 2 ATP was there. 2 NADH plus H plus was produced from glycolysis alone. In oxidative decarboxylation, there was no ATP produced, but per pyruvic acid, there was 1 NADH produced. For 2 pyruvic acid, you will have 2 NADH produced. In Krebs cycle, per cycle of pyruvic acid, per cycle of acetyl CoA, You had 3 NADH produced. So if there are 2 acetyl-CoA, there will be 6 NADH. Per cycle it is 1 GTP. So 2 cycles, because of 2 acetyl-CoA, it will be 2 GTP, and FADH2 number also will be 2. Is it clear? Nitrogen cycle is not even part of your syllabus. Okay. So totally from all of this combined so far, we have 10 molecules of NADH plus H plus, 2 molecules of FADH2, 4 molecules of ATP, and 6 carbon dioxide. All the carbon dioxide molecules are released. Okay. So now we have to work with these two. This can be directly used. This cannot be directly used. This has to go through ETS. So let's solve some questions based on Krebs cycle before we learn about ETS. During aerobic respiration, which of the following conversions shows substrate level phosphorylation? First, understand where substrate level phosphorylation, what does that mean? Wherever GTP is taking place, there substrate level phosphorylation will happen. Where did substrate level phosphorylation happen? Arnav is saying C, Vishal says C, Monica B, Elizabeth C. We just discussed it, right? Where did I say GTP is forming? This is substrate level phosphorylation. I mentioned whenever succinyl coenzyme is getting converted into succinic acid, a molecule of GTP will form. That is our substrate level phosphorylation. So the correct answer to this question is option C. Only 21% of you all have answered correctly. Vishal has answered in less than 10 seconds. Brilliant. That means you've completely memorized the cycle. Nani and Sagar, they have answered. I don't know what about the rest of you all. Here is another question based on Krebs cycle. श्रेयस, यहाँ पे वैसे भी लोग हैं जिन्हें हिंदी समझ में नहीं आता है. हाँ, कि कई लोग तमिलियंस भी है, कन्नडिगास भी है. तो इंग्लिश, इस चैनल का नाम ही फिजिक्स वाला इंग्लिश है ना, तो यहाँ पे इंग्लिश में ही, प्योर इंग्लिश में ही पढ़ाना जाता है. पढ़ाया जाता है. Okay. Choose the incorrect statement. You have to find out which is the incorrect statement. I'm not seeing a lot of answers. I wonder why not many people are participating. Shahil says A. The first product of Krebs cycle is a 6-carbon compound formed by condensation. That is true. A single turn of Krebs cycle produces 1 FADH2, 3 NADH2 and 1 GTP. Correct. All enzymes of the cycle are present in the matrix of the mitochondria. Is it? Is it? Not at all. Not all of them are present in the mitochondria. Succinate, dehydrogenase. It is located where? In the inner mitochondrial membrane. Option D, acetyl-CoA, high energy compound, acts as the substrate entrant for the cycle. Yeah, that is the entrant. That is also correct. So the incorrect statement with respect to Krebs cycle is option C. Just 33% of you all have answered correctly. What is the doubt? Is it clear now after I explained? What is the problem, Rohit and Sagar? Oh, there's a lag in the chat box. But did you answer correctly? What is your doubt, Monica? Did you not understand the question? Option D, you did not understand properly. Okay. See, in Krebs cycle, we start off with oxaloacetic acid condensing with acetyl coenzyme A to form citric acid, right? So, Krebs cycle, acetyl-CoA is entrant for Krebs cycle. Acetyl-CoA is an end product of link reaction. That is what is entering the Krebs cycle. That is what it means. It is true, right? Understood, Venkat Pushpa? Okay. Now let's take a look at our next question. The enzyme responsible for condensation of 2 carbon compound with 4 carbon compound in TCA cycle is citrate synthetase, succinic dehydrogenase, fumarase, isocitrate dehydrogenase. Sweetie saying B. Many are saying A. Why Sweetie and Anjali think B? I do not know. Option A is the correct answer. The enzyme responsible for condensation of 2 carbon compound. Which is this 2 carbon compound? It is acetyl coenzyme A. And with the 4 carbon compound. Which is the 4 carbon compound? It is our oxaloacetic acid. What does it form? It forms citric acid. For formation of citric acid, which is the enzyme required? Citrate synthase. Option A is the correct answer. Had I not run a poll for this question? My bad. Here's our next question. Which enzyme which acts as a connecting link between Krebs cycle and ETS is? NAD-linked malate dehydrogenase, FAD-linked succinic dehydrogenase, NAD-linked succinic dehydrogenase, FAD-linked isocitrate dehydrogenase. Many of you all are saying C. Sagar says B. Pratik also says B. Arnav thinks it's A. What did I tell you when I spoke about succinate dehydrogenase? What does it do? It does two things, right? It will catalyze the oxidation of succinic acid to fumaric acid. Simultaneously, it will catalyze the reduction of FAD to FADH2. So, is it FAD linked or NAD linked? Of course, you got succinate dehydrogenase correct. But is it FAD linked or NAD linked? It's FAD linked, right? So, the correct answer to this question here is option B, not option C. Many of you have got it wrong. You just had to think properly for like Daksab, what name is, I don't know if I'm pronouncing it correctly. Then we have Naga and Sagar who answered it correctly. Why? Have you got it clearly now? Have you understood it? It's FAD linked, not NAD linked. Succinate dehydrogenase, succinic dehydrogenase, same thing, no issues here. But it's linked to FAD. Next question. It's a lengthy question. Options are very long. Which statement is wrong for Krebs cycle? Which is wrong for Krebs cycle? Read carefully. There are 3 points in the cycle where NAD plus is reduced to NADH and H plus. There's 1 point in the cycle where FAD plus is reduced to FADH. During the conversion of succinyl-CoA to succinic acid, a molecule of GTP is synthesized. The cycle starts with the condensation of acetyl group with pyruvic acid to yield citric acid. D is incorrect. Everyone's saying, who can tell me why D is incorrect? What is incorrect about D? What is incorrect about D? Who can tell me? Abhijit, we'll learn about that when we go to ETS. Right now, we are only focusing on Krebs cycle questions. Acetyl-CoA part is correct. Pyruvic acid is wrong. Instead of pyruvic acid, it's oxaloacetic acid. 2-carbon acetyl-CoA along with 4-carbon oxaloacetic acid will give us a 6-carbon citric acid. Right? That is why this is incorrect. Next question. Identify the given reaction. It's starting with pyruvic acid. In the process, acetyl-CoA is produced as a product and also carbon dioxide is released. Decarboxylation is happening. Oxidation is happening. What is it called? Okay, majority of you have answered it as A. A itself is the correct answer. This is oxidative carboxylation. Who is going to tell me the alternative name for oxidative carboxylation? Oxidative carboxylation is also called? Yes, Abhijit says link reaction, correct. Next question. Read the following statements. What are they related to? Accept a molecule is a 4-carbon containing organic acid. First step is condensation reaction. It involves a single step of substrate level phosphorylation. There are 2 steps of decarboxylation. Most of you have answered correctly. Soumya is saying B. 2 steps of decarboxylation. First acceptor molecule is a 4-carbon containing compound. In link reaction, 4-carbon containing compound itself is not there, right? So the correct answer is TCA cycle, also known as Krebs cycle. So the correct answer is option C. Dr. Saab, what's your real name? And there's a kitty cat here. And Sagar, Arnav, Akash, Naga and Vishal, all of you answered correctly. Congratulations. Next question. In which of the following reactions of Krebs cycle substrate level phosphorylation is taking place? I will skip this because we've already done it. Substrate level phosphorylation happens only in one step, and that is in succinyl-CoA to succinic acid. We already discussed that in another question, right? Oh, but you still got it wrong. Who's saying B? Okay, this is yeah, option B is the correct answer. Renuka is saying C. Phosphoenol pyruvate to pyruvic acid substrate level phosphorylation will take place, but that's not the part of Krebs cycle. In Krebs cycle, it happens only in option B. In Krebs cycle, only one substrate level phosphorylation will take place. Right? Did you understood? Renu Kumari, Kurmi. Here, substrate level phosphorylation will take place, but that's not a part of Krebs cycle. So only B is correct. Arnav, Vishal, Naga, Sagar, Anjali are on the top 5. Next question. In TCA cycle, how many reduced coenzymes are produced from one acetyl coenzyme A? How many NADH and FADH2 are produced from one acetyl CoA? Yes, option A is the correct answer. Very good. Okay. So with that, we have finished Krebs cycle. Let's talk about electron transport chain or the electron transport system that exists in the inner mitochondrial membrane. So if you think of this as the mitochondria, you have your outer membrane which is smooth. Inside that you have the inner membrane which is kind of has many infoldings. These are called cristae. So far, whatever link reaction took place and your Krebs cycle took place, all of that happened here. Whatever NADH and FADH2 are produced, they are present here. Okay, so here you have NADH and FADH2. Whatever we obtain from link reaction and Krebs cycle. This has to go to the inner mitochondrial membrane for getting ATP. Okay, so within the inner mitochondrial membrane is where we have the ETC or your electron transport system. So if you take a look at this portion here, this membrane, here, if you focus here, you can see that on one side of the membrane, towards the inner side of the membrane, you have the matrix. And at the matrix, you have NADH plus, and FADH2. And on the other side of the membrane, you have the intermembrane space between the two mitochondrial membranes. This is your intermembrane space. Similar to all your membranes in your cells, even this inner membrane will have a phospholipid bilayer. You know the structure of plasma membrane, right? You've learnt about the fluid mosaic model. In the fluid mosaic model, you know that there are phospholipids and there will be protein complexes. There will be certain complexes present in the membrane. Very similarly, within this membrane, there will be 5 different types of complexes. In each complex, there are many different components. So whatever electron is present in NADH and FADH2, it will be passed on to this series of electron carriers present in these complexes. As the electrons are getting passed, ATP will get synthesized. Okay, so that is what happens in electron transport system. This is also called oxidative phosphorylation. Understood? This is the difficult portion of the chapter, but hopefully after this session, you will never say it is difficult again. Ramesh, hi, hello and all, you should not do here. This is not the platform for that. For socializing, find some other place. If you're here to learn, good. Otherwise, please leave. That is also a complex. It is not involved in electron transport, but it is a complex that is present. What do we mean by a complex? More than one things are present. If you see ATP synthase, that is also complex. It has F0, F1, 2 particles, right? 2 components. So that is a complex, but it's not involved in electron transport, but it's present within the inner mitochondrial membrane itself. Right, Abhijit? Understood? Okay, Moses is asking for the difference. See, here you have mitochondria. Mitochondria has 2 membranes. This is your outer membrane which is smooth. We don't care about it for now. What we need right now is present in the inner membrane. Inner membrane is this membrane that I'm talking about. If you look at this inner membrane, on one side of this membrane you have the fluid matrix inside. What is present on the other side? On the other side, there is space between your inner and outer membranes. This space is called intermembrane space. Inter means in between. In between the two membranes, whatever space is there, that is your intermembrane space. Okay. So you have inner membrane. Within the inner membrane, you have the 5 complexes. On one side of the inner membrane, towards the inside, you will have the matrix. Towards the outside, you will have the intermembrane space. Now, where is all the NADH and FADH2 located? It is present towards the inner side of the membrane, which is your matrix side. Is it clear? Moses, did you understand? Are you there? Okay, I hope you've understood. Okay. So, whatever electron gets passed on from one electron carrier to another, that entire pathway is what we refer to as your electron transport system. Where does this take place? Very clearly, it's very obvious, it's taking place in your inner mitochondrial membrane. What are some components involved? I told you there are 5 complexes involved. The 5 complexes are called complex 1, 2, 3, 4, 5. It's not very simple. Each complex has its own fancy name. We'll learn about it in just a bit. Okay. So there are 5 complexes. 1, 2, 3, 4, 5. Okay. Next, what are some of the characteristics? Over here, a series of redox reactions takes place. What does that mean? Whenever electron transfer is involved, whenever an electron is transferred, that molecule becomes oxidized. Whatever is accepting electron will become reduced, right? So if something is getting oxidized, another thing will get reduced. Over here, a series of such reactions will take place. That is what is the characteristic feature of electron transport chain. And whatever energy is stored in NADH and FADH2, that energy will get released in the form of ATP, which the cell can directly use. That is what is going to happen here. Any doubts so far? Any doubts so far? It's not O, it's not F naught, F0. F naught, F1. Abhijit, when we come to that, I'll tell you. Okay. So right now, in your NCERT textbook, this diagram is given vertically like that. I was not able to fit it all in like this, so I've done it horizontally, but it's the exact same thing, no difference absolutely. Whatever is there vertically, I've made it horizontal. It's the same diagram. I think I've used the same colors also. Okay. Now, what you're seeing in this diagram, pay attention, please don't spam. What you're seeing in this diagram is the membrane. This thing here is your phospholipid bilayer of the inner membrane. You know that within the phospholipid bilayer, there will be many protein complexes present. So 1, 2, 3, 4, 5, these are your protein complexes. First, let us understand what are the different complexes that are present in each complex and what is the fancy name of each complex. Okay. So the first complex, complex 1, is called NADH dehydrogenase. Why is it called NADH dehydrogenase? It will accept the electrons from NADH2. It will accept the electrons from NADH. That is why it is called NADH dehydrogenase. That is the name of your first complex. Within the first complex, there are 2 components. One is FMN. FMN is short for flavin mononucleotide. Along with that, there is an iron sulfur protein that we represent as FES. Complex 1, NADH dehydrogenase, that will receive electrons from NADH. See, matrix is here. In the matrix, NADH is there. That will provide electrons to this. Within the first complex, you have 2 components. FMN, which is flavin mononucleotide, and FES, which is iron sulfur protein. Okay, so that is the first part. Second complex. Okay, one other thing. Please notice how this is not in the correct sequence. You have 1, 3, 2, 4, 5. Why it is so, I'll tell you later. For now, let's take a look at the second complex here. The second complex is called succinate dehydrogenase. Where have we learnt about it? Succinate dehydrogenase is the enzyme that catalyzes the formation of FADH2 during Krebs cycle. And I told you that is the link between Krebs cycle and ETS, and that is the enzyme that's located in your inner mitochondrial membrane. So it's the exact same succinate dehydrogenase. FADH2 will donate electrons to this complex. Okay, so this is succinate dehydrogenase. Within this, again, you have 2 components. FES is common to this as well. Iron sulfur protein is there here as well. Along with that, you have FAD. What is FAD? Flavin adenine dinucleotide. FAD, flavin adenine dinucleotide. Okay, FMN is flavin mononucleotide. FMN complex 1, FAD complex 2. Very easy to remember. This is obtaining electrons from FADH2, right? So this is FAD. Here you have FMN. In both of them, iron sulfur protein is there. Clear? Now let's talk about the third complex. The third complex is called cytochrome BC1 complex. You've learnt about cytochrome B6F complex in photosynthesis, right? In cytochrome B6F complex, you have cytochrome B6 plus cytochrome F. Over here in cytochrome BC1 complex, obviously you will have cytochrome B, obviously you will have cytochrome C1, but along with that you will have iron sulfur protein as well. So if you look at complex 1, 2, 3, what is common? Iron sulfur protein is common. What is our fourth complex? Fourth complex is cytochrome C oxidase. Within the fourth complex, you do not have iron sulfur protein. You will have 2 cytochromes and 2 copper centers. 2 cytochromes are cytochrome A and cytochrome A3. The 2 copper centers are copper A, copper center A and copper center B. Okay, so this is your fourth complex. First complex, NADH dehydrogenase. Second complex, succinate dehydrogenase. Third complex, cytochrome BC1 complex. Fourth complex, cytochrome C oxidase. Fifth complex is ATP synthase. Within the fifth complex ATP synthase, there are 2 parts, F0 particle and F1 particle. This is called Racker's particles or oxysomes also. You may have learnt about this in the chapter Cell the Unit of Life when you studied about mitochondria, but even if you don't know, it's alright. So it is through this molecule that ATP will get synthesized. Alright, so you have F0, F1 particle. These are the 2 components. Is it clear so far? These are the different components in the different complexes. Is it visible clearly on the screen? Are all the parts visible clearly? Complex 3 name. Cytochrome BC1 complex. Yeah, see, right now we're doing so many things that require energy, but it takes so much to produce 1 ATP. When we get to that, I'll tell you, Anshul. Okay. So right now, first let's learn about how NADH will generate ATP. Next, we'll focus on FADH2. Okay. So let me first duplicate this once. Yeah. So first we'll focus on the route NADH takes. Where is NADH present? NADH is present in the matrix. So it's present in the matrix side. This NADH in the matrix will become oxidized and donate the electron to the first complex. Within the first complex, you have 2 components. The first component is FMN, the second component is iron sulfur protein. So the electron from NADH will first be given to oh my god, what did I do? Yeah, will first be given to FMN, and from FMN, first to FMN, and then from FMN it is given to FES, which is your iron sulfur protein. From FES, it has to be transported or transferred to complex 3. From first complex, there is no second complex, directly it's transported to complex 3. But as you can see clearly, there is a distance between complex 1 and complex 3. You can't just play catch like that with electrons, right? So there has to be something that picks up this electron from complex 1 and goes and gives it to complex 3. In order to do that, that molecule has to be mobile. That should be able to move from one place to another. Complex 1 and complex 3 are stationary. They cannot move within the membrane. But an electron has to be transferred from complex 1 to complex 3. There should be some delivery partner that picks it up from one place and gives it to the other, like our Dunzo and Squeaky, right? So for that, ubiquinone is helpful. Ubiquinone is very similar to the plastoquinone that you learn about in photosynthesis. This is a mobile electron carrier that is present right within the inner membrane. This is our inner membrane, right? This is our inner membrane. This is our inner membrane. Right within the inner membrane, this ubiquinone is present. Now, this ubiquinone will accept the electron from first complex. It will become reduced and it will become ubiquinol. Ubiquinol is UQH. That shows that it's becoming reduced. Okay. So, ubiquinone becomes ubiquinol. It gets reduced to ubiquinol. and then it will transfer that electron to the third complex. So as electron is getting transported through the first complex via the ubiquinone to the third complex, per NADH, 4 protons are pumped from the stroma side to the from the matrix side to the intermembrane space. Rohit, yes, coenzyme Q. Yes, it is also a proton carrier. Along with electron from here, it will pick up a proton from the matrix. You're right, and it will pump it inside. Right. So yeah. So what happened so far? From NADH to first complex, from the first complex to ubiquinone, which is a mobile electron carrier. Ubiquinone will accept the electron, it will pick up the protons from the matrix and it will get reduced to ubiquinol. Then it will donate the electron to the third complex. In the third complex, you have various components. How exactly do these electrons get transported? First, it will get transported or transferred, in fact, to cytochrome B. Cytochrome B to iron sulfur protein. From the iron sulfur protein to cytochrome C1. Okay, so this is how it will get transported. From the cytochrome C1, it further has to get transported to complex 4. Over here, complex 3 story is over. Now this electron has to be transported to complex 4. Similar to how there was a distance between 1 and 3, there is distance between 3 and 4. If electron from here has to be transferred to complex 4, we need another mobile electron carrier. Where is that mobile electron carrier present? This time it's not present within the membrane, it's present on the outer side of the inner membrane. Remember the location very clearly. This is the membrane, this is your inner membrane, this is the inner side of inner membrane, this is your outer side of inner membrane. Cytochrome C, look at how both of them are mobile electron carriers, look at the difference in where they are located. Ubiquinone is present within the membrane itself. Cytochrome C is present on the outer side of your inner membrane. So this cytochrome C will carry the electron from cytochrome C1, move along because it is mobile and pass it on to complex 4, which is cytochrome C oxidase. In complex 4 again you have 4 components. You have 2 cytochromes and you have 2 copper centers. How will it get transported here? From cytochrome C to copper center A, then to cytochrome A, then to cytochrome A3, then to copper center B. While electrons are being transported through the different components in complex 4, again protons will get pumped from here to here. Over here, whenever NADH is involved, you can see via complex 1, 4 protons are pumped from matrix side to intermembrane space. Via complex 3, 4 protons are pumped from the matrix side to intermembrane space. Via complex 4, 2 protons are pumped from the matrix side to the intermembrane space. Why am I saying pump, pump, pump here? Why am I not saying generally transport? Why I am saying pump is because pump is a type of active transport where you already have more protons here. The concentration of protons is already greater compared to here. Despite that, more protons are making use of energy and pumping it from this side to this side. Why is it happening that way? Because we need energy for ATP synthesis. As we move on, you'll understand. Alright. So right now from 1 NADH, 4 plus 4 plus 2, totally 10 protons are pumped from the matrix side to the intermembrane space. This has resulted in an electrochemical gradient across the membrane. What do we mean by electrochemical gradient? You may have learned about this in neural control and coordination, chapter, where you have the membrane is polarized, right? Very similar to that, even over here you can see one side of the membrane is more positive, the other side of the membrane is more negative. Why is this side of the membrane more positive? Because obviously this has a higher quantity of protons. Protons are positively charged, so here it is more positive. Outside it is relatively less positive. So it's more negative here, more positive here. Now, there are more protons here. In order to bring about equilibrium, these protons will have to move out so that on either sides, the same amount of protons are present. In order to do that, these protons cannot randomly diffuse through the plasma membrane. They cannot diffuse through this phospholipid bilayer because they are charged particles, right? If they are polar, if they are charged, you cannot move through the plasma membrane's phospholipid bilayer directly. So what is the other pathway that it has? It will go through your F0, F1 particle, which is your ATP synthase. What is extrinsic? Yes. Whenever we are saying pump Shahil, it will require ATP. Okay. So we have a total of 10 protons pumped from the matrix side to the intermembrane space. Right now, in the ATP synthase, I told you there are two components, F0 particle and F1 particle. This F0 component is present within the membrane. It's a transmembrane protein channel. It forms a channel, it forms like a tube-like thing through which the protons can move. This F1 particle is actually the ATP synthase head. When ATP synthase has to be synthesized, ATP molecule has to be synthesized, the enzyme that catalyzes the synthesis of ATP is your F1 particle. This is a peripheral protein. It extends out of the membrane. You can think of it like a lollipop kind of structure, where whichever is the transmembrane protein, that is like a tunnel, it is like a channel through which the protons will pass along its gradient. From where it's more to where it's less, it's going to pass like this. As the protons are passing like this, the ATP synthase head will rotate. Because of some conformational changes, it will synthesize ATP. Okay. So this F1 particle, you can imagine it to be like a peeled orange. It has many different subunits. Okay. So this F1 particle will have three conformations majorly. One is the loose conformation, one is the tight conformation, one is the open conformation. What happens when it is in the loose conformation is, it will hold on to one ADP and one inorganic phosphate. Then it will combine that into ATP. Then it will release that ATP. So every time this is rotating, the head is rotating, ATP will get synthesized. For the synthesis of one ATP, actually this is where there is a difference in your old textbook and your new textbook. In your old textbook, it was given that every 2 protons are required for the synthesis of one ATP. But newly it says in the text that 4 protons are required for one ATP, but in the diagram it still shows 2 protons only. So what happens here is, as the protons move through this channel, ATP will get synthesized. According to the new update in the text region of your textbook, if you consider 4 protons are required per ATP, we know that one NADH, we got 10 protons. If per ATP you need 4 protons, then from one NADH you get 2.5 ATP. You can round it off to 3. Actually, this is very controversial. There are so many books that give different numbers, but I am giving to you the number according to the update that you have in your newest rationalized. I don't know what you're talking about, Rohit. I don't know what it is. In Neet rationalized syllabus, they have made the update in the text, but the diagram still they are showing 2 protons only. But according to the text, if you see per NADH, you'll get 2.5 ATP. You can kind of round it off to 3. That is how you can relate NCERT. But you have so many different sources on so many different websites, even top publishers of biochemistry. Different things have different sources. It's still undergoing a lot of research. So let's not get confused at this point and focus on what is given in NCERT, because the questions will be based on that. Alright. So per NADH will get around 3 ATPs. Through complex 1, 4 ATP. Through complex 3, 4 ATP. Through complex 4, 2 ATP. Totally, I'm sorry, am I saying ATP every time? It's protons. 10 protons are pumped, and from 10 protons we will get around 2.5 to 3 ATP. Okay, so this is the story of NADH. Wait, I forgot something. Yeah, so finally from complex 4, it goes to copper center A, cytochrome A3, complex B. Ultimately, the electron is donated to oxygen. Oxygen is the final electron acceptor. Okay, so far we've been saying aerobic respiration, we didn't talk about it in glycolysis, we didn't talk about the involvement of oxygen in link reaction or Krebs cycle. Finally, oxygen's role is in ETS. So this will accept electrons from copper center B, pick up a couple of protons from the matrix and form water. Okay, so this is the story of NADH. Now let's talk about FADH2. In FADH2, complex 1 is not involved. You know complex 2 is succinate dehydrogenase, right? So FADH2 for formation also this has catalyzed. So FAD will donate the electron to this complex. From this complex, very similar to how ubiquinone was the mobile electron carrier in complex 1, even here ubiquinone, which is present in the membrane itself, will pick up the electrons from iron sulfur protein in complex 2, pass it on to the same complex 3. As the electron is getting transported through the different electron carriers in complex 3, 4 protons will be pumped from the matrix side to the intermembrane space side. Then the same story continues. Then through cytochrome C, it goes through complex 4. Finally, from complex 4, it is donated to oxygen. And oxygen combines with protons to form a molecule of water. Okay, so over here if you see, in NADH, whenever NADH was donating the electrons, complex 1, complex 3, complex 4, all of them were pumping protons. So totally 10 protons got accumulated in the intermembrane space. But if you look at the story of FADH2, complex 2 is not a proton pump. It does not pump any protons. In complex 3, you have 4 protons pumped. By complex 4, you have 2 protons pumped. So from one molecule of FADH2, you will have 6 protons pumped. So if 4 protons are required for 1 ATP, then from 6 protons, you get around 1.5 ATP, which we kind of round off to 2 ATP. So from one FADH2 molecule, you will get 2 ATPs. Is that clear? Abhijit, what is the name of the proton pump in complex 1? Can you please tell the mechanism by which it pumps? It's beyond the scope of your syllabus, so I don't think you should... Yes. Anshul, terminal acceptor is oxygen. Up until this point, oxygen is not involved. Is it clear so far? Venkata Pushpa, are you still there? Soumya, you're asking about the Neet syllabus. Neet syllabus, you should look at NMC, whatever syllabus is updated by NMC website. According to that, you have to go. For example, if you take the chapter Living World, in the new syllabus, what is living part is not there. If you take organisms and population chapter, organism and environment, that chapter, that portion is deleted from the new syllabus, but it's still there in your old syllabus. Okay, so you have to take a look at the NMC updated syllabus and study accordingly. Venkata Pushpa Harika, have you understood? You still think it is the toughest? All right. So from the electron transport chain, if you consider NADH, first complex 1 to complex 3 to complex 4. Why is there a complex 5 here? From complex 4 to oxygen. From one NADH, we get approximately 3 ATP. From one FADH2, you get approximately 2 ATP. Okay. So we have to find out under ideal conditions how many ATP can be produced from one molecule of glucose under ideal conditions. That is our respiratory balance sheet. If everything, yeah, we'll consider what are the assumptions that we make. Okay, so if we have to calculate from one molecule of glucose how many ATP is getting synthesized, we have to make certain assumptions. What are those? First of all, there is sequential orderly pathway where whatever glucose we are starting off with, it is going to go through the exact steps of glycolysis. None of the metabolites, none of the intermediates are withdrawn for other things. That is one assumption we're making. Whatever pyruvic acid is forming, that will go into the mitochondria and it will undergo the oxidative decarboxylation. It will enter Krebs cycle. Everything is happening in sequence. That is one assumption. Secondly, whatever NADH was synthesized in glycolysis 6th step, that we will assume that it will be transferred into the mitochondria and that will also undergo oxidative decarboxylation. The third point is, yeah, I already mentioned this, whatever intermediates are produced in any of these steps, be it glycolysis or Krebs cycle, any of these steps, none of these intermediates are utilized to synthesize any other compound. They are not withdrawn from this to synthesize anything else. That is the other assumption. And finally, only glucose is being respired, no other alternative substrates are being respired, and none of them are entering the glycolytic pathway in between anywhere. If you make these assumptions and calculate how many NADH are produced, how many ATP are produced, we will get our I doubt that, Abhijit. I'll check anyway. Ramesh Verma, please. This is not the place to socialize. Behave yourself. Yeah. So now let's take a look at the respiratory balance sheet. From glycolysis, we got 2 ATP, 2 NADH. From each NADH, if you can get 3 ATPs, around 3 ATPs, that is the approximation rounding off. So per NADH, if you can get 3, so totally from 2 NADH that we got from glycolysis, we will be getting a total of 6 ATP. Okay, so 2 ATP directly substrate level phosphorylation. From oxidative phosphorylation, we get 6 ATP. So from glycolysis, totally we are getting like 10 ATP. Pyruvate oxidation, or basically oxidative decarboxylation, if you say, over there no ATP was produced. Per pyruvic acid, 1 NADH was produced. Since we have 2 pyruvic acid, 2 NADH will be produced. Per NADH, if it's 3 ATP, totally 6 ATP is produced from our link reaction. After that, in Krebs cycle, per acetyl CoA, you have 2 GTP or 2 ATP produced. So 2 ATP there. Per NADH 2 ATP, we have a total of 6 for 2 acetyl coenzyme A, that is 18 ATP there, and 2 FADH2 that was produced. From that you will get 4 ATP. So totally 18, 20, 24. 24 ATP here, 6 ATP here, and 10 ATP here. Where am I going wrong? 6, 7, 8. This is 8, no? Why did I make it 10? So totally you will get 38 ATP is the net yield of glucose. Is that clear? Okay, cool. Now let's solve a couple of questions. Why don't you want to solve questions, Abhijit? What's the hurry? I have some very interesting questions added. Select the incorrect statement. Glycolysis occurs in the cytoplasm of all living organisms. Yes. Enzymes of Krebs cycle are present in the mitochondrial matrix. Are they? Oxidative phosphorylation takes place in the outer mitochondrial membrane. Huh? Through link reaction, pyruvate enters the aerobic respiratory pathway. Correct. But enzymes of Krebs cycle, not all of them are present. Of course, they are not mentioning all. Yeah, some of them are present. So if you consider these two, this is like absolutely wrong, right? Oxidative phosphorylation, where does that take place? It takes place in the inner mitochondrial membrane, not outer. Krebs cycle enzymes, most of them are in the matrix. Succinate dehydrogenase is in the inner mitochondrial membrane. But however, since they have not mentioned all, you can see C can be marked as the correct answer. Next question. Which of the following elements combines with protons to form metabolic water during ETS? Yes, B is the correct answer. Oxygen is the final acceptor of electron and protons from the matrix to form water. Naga, Sujal, Nani, Rohit, Sai Prem, Akash, Harika, everybody has answered correctly. Here's our next question. In electron transport system of respiration, which enzyme complex is odd with respect to electron carrier? Naga has answered very quickly. Sujal also. But let's see if it's correct or wrong. Why is it odd? Who can tell me? Awesome. Naga, why is it odd? What is odd about complex 5? Who can tell me? Why is it wrong? It forms ATP, right? But it is not part of the electron transport system, right? None of its components are involved in electron transport. Next question. Maximum energy is released during the conversion of. Maximum energy is involved in, is released in during the conversion of glucose to pyruvic acid, 2 ATP to NADH, glucose to ethyl alcohol and carbon dioxide, 2 ATP only. No, wait. Yeah, 2 ATP only. Pyruvic acid into acetyl coenzyme A, 1 NADH is produced. Pyruvic acid into carbon dioxide and water. Option D is the correct answer. That is nothing but your ETS. It is in the ETS that the maximum number of ATP is formed. Why is it that only few people have answered correctly? Why do you think? What is the confusion? Next. During ETS, the terminal electron acceptor is. Terminal electron acceptor is. Everybody is saying C. My son has come back from school. Can you hear him scream? Everyone has answered correctly. Now, I'm going to leave a couple of questions in your notes for your practice. Okay. Last two topics are left. We'll finish that off. What is amphibolic pathway? We all know that there is anabolic pathway and catabolic pathway. Anabolic pathway is when something is getting built up like photosynthesis. In photosynthesis, what happens? Carbon dioxide has 6 molecules of carbon dioxide come together to form 1 molecule of glucose, right? So something small is getting built up to form something bigger. That is your anabolism. Generally, anabolic reactions require energy. What are catabolic reactions? Glucose, which is a large molecule having 6 carbons. Right now, as we are seeing in cellular respiration, it gets broken down to produce 6 molecules of carbon dioxide. From one big molecule, you're getting many smaller molecules. So during such processes, energy will get released. This everybody knows, anabolism, catabolism. Amphi means what? Amphi means both. Amphitrachus you've learnt about in bacteria. When on two ends also there is flagella, we call it amphitrachus. Amphi means both. So what is amphibolic? Generally, respiration was thought to be a catabolic process where glucose is getting broken down. But if you take a closer look, it's not like a Google Maps thing that you go here, you take a right, you take a left, right? After this has to happen, this has to happen. All of these reactions, for example, if you consider glycolysis, all of these reactions, all of the 10 reactions are taking place in the cytoplasm. It's not necessary that the glucose 6-phosphate that is formed has to form fructose 6-phosphate. Glucose 6-phosphate can go do something else also, because it's not like a restricted thing. They do not have instructions that after this has to happen. Okay, so it's not like every one of these molecules have brains and they know what to do next. These are random chemicals that are interacting in the cytoplasm. So when such is the case, when glucose is getting broken down, carbohydrates such as glucose is getting broken down, there are possibilities where some of the intermediates in glucose, in glycolysis, can get removed from the glycolytic pathway, which is a breakdown pathway, and get included in some pathway where building up is happening. So right now in glycolysis, if you think catabolism is taking place, glycolysis is breaking down of sugar is happening, breaking down of glucose is happening. Some of the intermediates present here, for example, pyruvic acid, that is a product of glycolysis, it need not necessarily enter the link reaction or the Krebs cycle. If the cell at that point requires the synthesis of proteins, this pyruvic acid can be withdrawn from the glycolytic pathway and used in the synthesis of proteins. Pyruvic acid, after it will get converted into amino acids, and amino acids can be used in the synthesis of proteins. Likewise, generally the cell prefers glucose or carbohydrates to be the preferred substrate, but sometimes, like I mentioned earlier also, under certain conditions, fats and lipids and proteins also can act as respiratory substrates. In that case, they will not enter the pathway as glucose, right? So they will enter it somewhere intermediate. Fats have to first be broken down into fatty acids and glycerol. Fatty acids can get converted into acetyl-CoA and enter the pathway. If it's glycerol can get converted into glyceraldehyde, phosphoglyceraldehyde, and enter the pathway. If proteins are broken down, amino acids can enter the pathway as pyruvic acid or as acetyl-CoA after deamination. So that is possible. So what we're trying to say here is, initially, it used to be thought that respiration is a catabolic process, but if you look at it carefully, for the synthesis of proteins, pyruvic acid, which is an intermediate here, which is an intermediate metabolite, can be drawn from this cycle and used in the synthesis of something. When something is getting synthesized, it's anabolic. If it's getting broken down, it's catabolic. So the same intermediate in this pathway can be a part of catabolic pathway or an anabolic pathway. That is why it is called amphibolic pathway. Is this clear? Yeah, for homework, I've added some questions. It is possible to get 650 also, Nani, but it depends on how much work are you willing to put in, how prepared you are. It depends on you. Okay, it's possible to get 720 also. People have achieved that in the past, right? So if you're asking if it is possible, it's definitely possible. Yeah. So we've discussed fermentation, which is anaerobic respiration as well as aerobic respiration. What are some of the differences? Two differences are very clear to you. In fermentation, oxygen is not used. In aerobic respiration, oxygen is used. Second difference, in fermentation, not a lot of energy is produced. In aerobic respiration, a lot of energy in comparison is produced. In fermentation, it's 2 ATP, and in aerobic respiration, under ideal conditions, it's what? 38 ATP, right? So that's a huge difference. And also, in both cases, NADH that is produced will get, I'll get to that. In fermentation, there's only a partial breakdown, and in aerobic respiration, there is complete breakdown or complete oxidation. In fermentation, only 2 molecules are obtained, and in aerobic respiration, many more molecules are obtained, as many as 38 under ideal conditions. That is the second point. And if NADH has to get oxidized to NAD+, it will happen very slowly in fermentation, but in aerobic respiration, it happens very quickly. Okay, next, last concept, respiratory quotient. Respiratory quotient is basically, yes, I will try calling you Rohit, we'll try. Respiratory quotient is basically the ratio of, in respiration, in the process of cellular respiration, it depends on which substrate we have taken. Respiratory quotient is also called your respiratory ratio. It is the ratio of how much carbon dioxide has been evolved, how much carbon dioxide, how many molecules of carbon dioxide are released, and how many oxygen molecules are consumed. This ratio will give you your RQ. If you consider glucose, which is a carbohydrate, glucose has 6 carbon molecules. For glucose to completely be oxidized, we need 6 molecules of oxygen, and it releases 6 molecules of carbon dioxide. Since they are both equal, the respiratory quotient will be 1. If you take the example of tripalmitin, which is a lipid, you can see how the number of oxygen consumed or utilized is 145, while the number of carbon dioxide that is released is much lesser. In such a case, the ratio is going to be less than 1. It's about 0.7. For proteins, it could go up to 0.9. For some organic acids, it can be greater than 1 also. Okay? CO2, you have written superscript. It should be subscript, no Rohit. So yeah. Let's solve some questions. We have reached the end of the chapter. Last set of questions, and then we'll take a look at the leaderboard. Yeah. Slow oxidation occurs in fermentation. Yes, many of you are answering it as C, and C itself is the correct answer. All right. So that's it. There are some extra questions in this PDF that you will get based on electron transport chain. Of course, we could not find the time to solve all of them in today's lecture, but I request you all to try solving them. It will be in the PDF, but if you have any doubts in any of those questions, what you could do is you drop a comment in the comment section with the slide number, with which page it is on. I will answer your doubts. Okay. Intermediate of glycolysis to electron transport. Glycolysis to electron transport. I don't know what you're asking, Anshul. Let's take a look at the leaderboard. Akash, are you there? Akash L is on top of the list. He's answered 24 questions correctly. I'm sure there will be some glitches in the poll, and this is not like 100% accurate. But Akash, as always, he attends lectures. He's very attentive and he participates brilliant. Hopefully, you will get good marks. Over the time, I'm seeing how Naga has also improved one session after the other, and he's very consistent in attending and answering questions. Sagar also, Rohit also, Nani, Soumya, Venkata Pushpa also has been very regular. Anjali, Abhi, I think he was there in the last lecture as well. And Jatin, Arnav, all of you, thank you for participating. Hopefully, the session went a little over 2 hours, I think. I mean, 2 o'clock, I think. Yeah, it's 2:34 now. So, congratulations to all the people who participated. Hopefully, even if you got answers wrong, you have found out where exactly you're making mistakes, what exactly is the concept that you have not understood. Hopefully, you'll work on it. My next session will be for sexual reproduction in flowering plants. After this, you have plant growth and development, which Chaitra ma'am will be conducting. And after that, first chapter of second PU syllabus, sexual reproduction in flowering plants, I will be taking. It's the same for all subjects. There will be 2 teachers who will alternate. Okay. For the schedule, you have to check the community post tab. You will get, yeah, top 3 boys. Hopefully next class, there will be a girl also. Okay. You wanted to try the call thing, right? Let's try it. Rohit.
Hello.
Oh, hi. Can you hear Rohit?
Hello.
Hi, Rohit. Hello.
Hello, ma'am.
Hi. You're audible. Can everyone listen to Rohit?
Hello.
Can you hear Rohit? The rest of you? Maybe I should keep my mic here somewhere. Where are you from, Rohit? I can hear you. Can you not hear me? Oh, there's some random ad playing on my YouTube right now, and Rohit is gone. Rohit, can you not hear me?
Hello.
Please.
Ma'am.
Yes, Rohit, please somebody tell Rohit in the comment section that we can see and hear him. I don't think.
Hello.
Okay, this is not
Hello,
working.
ma'am.
Yes, Rohit, I can hear you. It's not working. Probably, I don't know what the issue is. I'll find out from some other teachers if they've ever used it. So, until I see you in the next class, don't waste any time. Study well. Aim for 720. Don't ask questions like, will I get 400, 300 and all that. Aim for the best possible and work hard towards your goals. All the very best. I'll see you soon in the next class. Bye bye.